नहीं सर प्लीज। बिस्मिल्लाहिर्रहमानिर्रहीम। अस्सलाम वालेकुम। Namaskar and a very good morning to the esteemed chief guest, Dr. Sanjay Kumar, Chairman, Agricultural Scientists Recruitment Board, Department of Agricultural Research and Education, Ministry of Agricultural and Farmers Welfare, Government of India. Honorable Vice Chancellor, Sir, Professor Dr. M. Afshar Alam, Professor S. Raisuddin, Dean Academics and Director IQAC, Professor Suhail Parvez, Dean, School of Chemical and Life Sciences, Professor M.Z. Abdeen, Director, Center for Environment and Sustainable Development, Dr. Humaira Farooqi, Head, Department of Biotechnology, esteemed guests who have graced us with their presence today, delegates, teachers, research scholars, and their students. I, Dr. Saima Wajid, feel privileged to welcome you all on behalf of Department of Biotechnology, School of Chemical and Life Sciences, Jamia Humdar, to the inaugural session of the DBT and DST SERB sponsored 3 days National Conference on Sustainable Biotech Solutions for Global Challenges. At the outset of the event, I would like to invite on stage Imam Sahab Rabia Mosque, Janab Farooqi Sahab, to recite a verse from Holy Quran and also its translation to enlighten us.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم لن تنالوا البر حتى تنفقوا مما تحبون। कुरान की एक बहुत छोटी सी आयत पढ़ी मैंने अभी आपके सामने। इसमें जो मैसेज है वो मैं अपनी ज़बान में आपके सामने रख रहा हूँ। और देखिए ये कितनी इम्पोर्टेंट बात कुरान ने कही है कि जब तक तुम अपनी किसी प्यारी चीज़ को कुर्बान नहीं करोगे उस वक्त तक तुम कामयाबी नहीं हासिल पा सकते। जो भी चीज़ तुमको सबसे ज़्यादा प्यारी है उसको तुम कुर्बान करो तब तुम अपनी मंजिल पे पहुंच पाओगे। अब ये हर आदमी का अलग अलग मामला है। किसी को अपनी ज़िंदगी प्यारी होती है, किसी को अपना वक्त प्यारा होता है, किसी को और कोई चीज़ प्यारी होती है। लेकिन अपनी कुछ प्यारी चीज़ कुर्बान कर दो फिर कामयाबी तुमको मिलेगी। ये बात मुझे इसलिए याद आ गई कि ये साइंटिस्ट जो हैं इनके यहाँ वक्त की बड़ी कीमत है। ये अपना वक्त बहुत ज़्यादा अपनी लैब में गुजारते हैं, बहुत ज़्यादा रिसर्च के कामों में ये अपना पूरी दिमाग की ताकत खर्च कर डालते हैं। फिर जो कुछ भी ये हासिल करते हैं उस वक्त की कुर्बानी देने के बाद तो वो इंसानियत के काम आती है। इस बात पर मुझे एक बात और आखिरी बात जो जिक्र कर रहा हूँ वो ये है कि 1847 तकरीबन 150-200 साल पहले की बात है। स्कॉटलैंड में एक साइंटिस्ट थे, उनका नाम था सर जेम्सन। वो अपनी लैब में बेहोश पड़े हुए थे और उनके 2 असिस्टेंट भी बेहोश पड़े हुए थे जमीन पर उल्टे मुंह। उनका नौकर अंदर आया, देखा क्या ये 3 आदमी बेहोश पड़े हुए हैं। वो समझा कि शायद तो शराब वराब पी ली है बहुत ज़्यादा इन्होंने, तो इसलिए ये बिल्कुल होश हवास में नहीं है। वो चला गया। वो दरअसल शराब वराब कुछ नहीं पिया था। इन्होंने क्लोरोफॉर्म जो इंसान को बेहोश करने की एक दवा है उसका तजर्बा अपने ऊपर किया कि अगर हम इसको नाक से अंदर डालें तो बड़े से बड़ा काम सर्जरी का हो सकता है इंसान को तकलीफ दिए बगैर। तो अपनी जिंदगी को दांव पर लगा दिया ताकि इंसानियत को कुछ दे सके। यही वजह है कि ये साइंटिस्ट जो हैं ये अपने घर का सुकून, अपने घर का आराम अपना कीमती वक्त, अपना खाना पीना, अपना रोज का रूटीन, सब चीज कुर्बान कर देते हैं। और उसके बाद फिर वो जो चीज देते हैं इंसानियत को, फिर वो इंसानियत के लिए बहुत बड़ा तोहफा होता है। अब ये सर जेम्स जो थे 1847 में जिन्होंने ये काम किया, अगर वो दवा ज्यादा ले लेते तो उनकी जिंदगी खत्म भी हो सकती थी, मगर उन्होंने कहा इसका तजर्बा मैं अपने ऊपर करूंगा, ताकि ताकि मैं कोई चीज बनाऊं और वो चीज जो है इंसानों के काम आए, इंसान कहीं कभी रहने वाला हो, किसी मजहब का मानने वाला हो, उसके काम आए। तो आज का जो मैसेज है वो यही है कि साइंटिस्ट जो हैं, कहीं भी दुनिया में किसी जगह भी हो साइंटिस्ट, ये अपनी बहुत बड़ी चीज कुर्बान करते हैं, और कुर्बान करने के बाद जो इंसानियत को देते हैं वो बस ऐसी चीज होती है कि उसकी कोई मिसाल नहीं मिल सकती। इसलिए मेरी दुआ है कि यहाँ जो साइंटिस्ट बैठे हैं, जिंदगी में हमेशा ये लोग तरक्की करें और दुनिया को ऐसी चीज देके जाएं कि जब ये दुनिया में ना भी रहें तो इनका नाम रोशन रहे।
Jazakallah Farooqi Sahab. I request the dignitaries on the dais to welcome our chief guest, Dr. Sanjay Kumar, with a planter and present him a memento and a shawl as a token of heartfelt gratitude. I request Professor Suhail Parvez, Dean SCLS, and Dr. Humaira Farooqi, Head Department of Biotechnology, to present a planter, a memento, and a shawl to the Honorable Vice Chancellor, Professor Dr. M. Afshar Alam, as a token of respect. He is a pillar of strength for us. May I request Professor M.Z. Abdeen and Dr. Humaira Farooqi to present a planter to Professor S. Raisuddin, Dean Academics and Director IQAC, Jamnagar. Planter to Professor Raisuddin, Dean Academics and Director IQAC, Jamnagar. I would request Honorable Vice Chancellor Sir and Professor Abdeen to present a planter and a memento to Professor Suhail Parvez, Dean, School of Chemical and Life Sciences. Professor Suhail Parvez, Dean, School of Chemical and Life Sciences. I would now request Honorable Vice Chancellor Sir to present. I would humbly request Honorable Vice Chancellor Sir to present planters to Professor M.Z. Abedin, Director CEST, and to Dr. Humaira Farooqi, Head Department of Biotechnology. Thank you so much. I would now like to call University Tarana team to present Hamdard Tarana. Tarana team to present Hamdard Tarana.
ये दर से गए हम दर्द है ये, हम इसके चांद सितारे हैं। ये दर से गए हम दर्द है ये दर से गए हम दर्द है ये। ताबीर है ये उस ख्वाब की, जिस ख्वाब की मंजिल हिकमत है, जिस ख्वाब की मंजिल हिकमत है। ये शम्मा ऐसी शम्मा है, जिसकी के जहां में शोहरत है, जिसकी के जहां में शोहरत है। इस वक्त जर्रे हैं हम सब, पर तारे बनकर चमकेंगे, मानिंद गुलों के महकेंगे, महताब की सूरत चमकेंगे। ये दर से गए हम दर्द है ये दर से गए हम दर्द है ये। पाई है यहाँ पर हमने जो दौलत बोला फानी है, दौलत बोला फानी है। हिकमत का पैगंबर था वो जो इस मेरे चमन का बानी है, इस मेरे चमन का बानी है। हर वक्त यहाँ पर हमने जो अखलाक मोहब्बत पाया है, हर समत जमाले इल्म यहाँ फूलों की तरह मुस्काया है। ये दर्स गए हम दर्द है ये दर्स गए हम दर्द है ये। नुसरत वो जफर का परचम हम इस आलम पर लहराएंगे, इस आलम पर लहराएंगे। इस रोशन शम्मा की खातिर हर तूफान से टकराएंगे, हर तूफान से टकराएंगे। सरसाज रहे शादाब रहे अल्लाह दुआ ये मेरी है, इस गुलशन पर ना आ जाए अल्लाह दुआ ये मेरी है। ये दर्स गए हम दर्द है ये दर्स गए हम दर्द है ये दर्स गए हम दर्द है।
Beautiful, Hakeem Janab Abdul Hamid Sahab, ये मेरे जन्म का बानी। The sustainable biotech solutions have emerged as a transformative force, offering innovative approaches to address pressing global issues as food security and nutrition, climate change, health-related challenges, etc. Biotechnology is driving the evolution of eco-friendly therapies and diagnostics, like biodegradable drug delivery systems. It is contributing in reducing carbon footprints. Sustainability through biomanufacturing takes into consideration the 3 R's: reduce, reuse, and recycle. The biotech solutions contribute to each and every sustainable development goals set up by United Nations. We expect that the deliberations in this conference shall be fruitful and enlightening for all the participants. I now request Professor Suhail Parvez, Dean SCLS, to kindly present the welcome address and share his thoughts on this occasion.
Assalamu alaikum and very good morning, everybody. It's an honor for me to present the welcome address. First of all, I would like to welcome the chief guest for today, Dr. Sanjay Kumar, who is the chairperson of ARSB. I would like to mention here, sir, when I was growing up as a kid, two memories have always etched in my mind. Bharat ek krishi pradhan desh hai, and in Doordarshan there was a program called as Krishi Darshan. And when I think back, when I reflect, the nice notion from our Imam Sahib regarding science and scientists, India has been there, and ASRB is promoting agricultural research for decades now, and this has gone to show that why we call it till today. So thanks a lot, sir, for accepting our invitation and letting us host you. It has been a wonderful setup, and we are waiting to hear from you. I would like to welcome our Honorable Vice Chancellor, Professor Afshar Alam, who in his tenure of almost 3.5 years has transformed everything. Only this week, the new global ranking has come up and we have gone one notch even higher. Every year, every year there is an increment, and we have now got our 10th FIST program sponsored by DST in the Department of Chemistry. Day by day, the citations, the publications, the grants, everything is being showcased in the NIRF ranking that we can see. So thank you, sir, for letting us organize these events where people from all across the country are coming and we are able to showcase. I would like to welcome Professor Raisuddin, who is a pillar of Jamia Hamdard. He has created IQAC in the past 10 decades. He created the RDC cell, and currently he is being entrusted with the role of being Dean Academics. And I'm very sure that that will be also a very big pillar in our university very soon. Thank you, sir, for gracing us today. I would like to welcome Professor Abdeen, a man with close to 3 decades of service to Jamia Hamdard, the man who resonates with the name of biotechnology, the man who created biotechnology out of nowhere. And currently, when we see the kind of demand that we have, none of the candidates sitting here who are in BSc biotechnology have a score in CU2 less than 140. So that's the department that is. So I would also like to welcome the passing on the baton from the older generation to the next generation, Dr. Humaira Faruqi, who is the current head and leading the department as the current head. And not only Dr. Humaira Faruqi, other senior faculty, Dr. Saima Wajid, and all others have put together a great show. I should share with you, I was part of the workshop that was inaugurated on Monday, and as I just said, that the star department of SCLS, everybody is looking forward to it, and I'm always very happy to be part of any events of that. So now from that to GM, genetically modified foods, then to crisis management of things that are currently termed as global crisis. So I would say biotechnology of Jaiv Takniki Shastra, as we call it, will definitely be the science in the next 1 decade to come for sure. And conferences like these, events like this, will definitely give us new perception and new landmarks to look for. Thank you very much. All the very best for the 3 days of the program that we are going to conduct. And I would congratulate the organizing committee, Dr. Monica, Dr. Javed, who have done a wonderful job in creating this. Thank you very much. All the very best.
Thank you, Professor Suhail, for setting a delightful tone for this conference. I'm enthusiastic to announce the conference abstract book launch as a souvenir by the distinguished chief guest, Dr. Sanjay Kumar, Honorable Vice Chancellor, Sir, Professor M. Afshar Alam, Dean SCLS, Professor Suhail Parvez, and the organizing committee members as the organizing secretary, Professor Abdeen, conveners, Dr. Humaira Faruqi, Dr. Jagriti Narang, and myself, co-conveners, Dr. Javed Ahmad Sheikh, Dr. Miraj Ahmad Ansari, Dr. Monica Safi, and Dr. Javed Ahmad. I would request the organizing committee kindly be on stage for the group photograph. Thank you, sir. It's time to introduce the theme of the conference to the august audience, and I invite Professor NZ Abdeen to present the introductory remarks.
Honorable Vice Chancellor, Professor Dr. Shah Sahab, the chief guest of today's inaugural session, Dr. Sanjay Kumar, Chairman ASRB, Government of India, the Dean Academics, Professor Raisuddin Sahab, Dean SCLS, Professor Suhail, HOD Dr. Mera Faruqi, the head of the departments, colleagues, delegates, students, good morning to all of you. Today, this is the inaugural session of the conference, that is national conference, that is Biotech Solutions for Global Challenges. The title itself suggests that how biotechnology is solving the problems that we are facing today. Actually, if we see the development of biotechnology as a subject, as an industry that begins with the discovery of DNA and its structure as well as function, and then central dogma of molecular biology was discovered, and ultimately that led to the foundation of biotech industry with the development of recombinant human insulin that replaced porcine and bovine insulin, which was having many side effects, some ethical issues, and that become a boon, recombinant human insulin, to the patients suffering from type 1 diabetes. Thereafter, actually, many developments occurred, and we can recall the BT cotton, and then there are many other BT crops which solve the problem of low productivity because of the manifestation of the pests and diseases in crop plants, including cotton. Thereafter, actually, scientists then started working on a nutritional aspect of the crop, biotech scientists, and now we know the developments that led to the development of a new rice variety that is referred as golden rice with vitamin A. And then we have bananas with vitamin A, and there are also crops which is having many minerals, micronutrients, for example, selenium enriched rice and wheat, and many more. And then actually now we have we are going to have crops that will be resilient to climate change, to increase in temperature. They may sustain increased temperature, then flooding, because we have flash flooding, and then actually we are going to have crops which will be resistant to many other biotic and abiotic. So actually the goal of the biotechnology is that we have to maintain not only the productivity but increase productivity because our population is growing, and then we have to also make these crop produce enriched with minerals and vitamins, so that they have better nutritional quality. Actually, as we already know that this conference is going to deliberate with the issues related to the change in the climate and how we can develop climate resilient crops. Then how we can also improve the nutritional quality of the produce that is coming, and then how we can improve the environment that is already degraded. Actually, we already know that there is a lot of change in the environmental conditions everywhere. As we all know that our AQI is very high, and then actually we have also global warming, and then their implications. So we need to have the biotech solutions which can improve these environmental conditions. Actually, we need to have like solutions to elevate metal toxicity, metal accumulation in the soil. So we have to we have to develop bioremediation processes which are cost effective, and then they can be used for this. Then we also need the processes where we can improve the nutritional imbalances in the soil. Actually, that has to be corrected so that we can sustain the productivity without further degrading the environment. We also need biopesticides to replace chemical pesticides because they will be harmful to us. So actually, these are things that we have to be done. We have to dwell with in the conference, and then we have to also see the health aspects. So actually, there are emerging diseases where pathogens have become multi-drug resistant. So we have to come up with the drugs that could actually that may be effective, very effective in these multi-drug resistant diseases. We have already seen biotechnology has come up with vaccine to combat COVID-19. So actually, because of the paucity of the time, I will not I will not give. So these are some examples that I have given, and that we have to discuss during the conference, and this conference will come up with some solutions and resolutions that will be then sent to government agencies for implementation. So this is all from me, and then I am handing over the dais to the organizers. Thank you very much.
Thank you, sir. May I now request Dr. Umaira Faruqi, Head Department of Biotechnology, to say a few words.
A very good morning to all. Honorable Vice Chancellor, Professor Mohammad Abshar Alam, our esteemed Chief Guest, Dr. Sanjay Kumar, Chairperson, ASRB, esteemed dignitaries on the dais and off the dais, distinguished faculty members, industry experts, young researchers, and their participants. I welcome you all, and it's my pleasure to stand before you, welcoming you to the 3-day national conference cum workshop on sustainable biotech solutions for global challenges. This event is a testament to the growing importance of biotechnology in addressing some of the most pressing challenges of our time, from climate change and food security to healthcare and environmental conservation. The Department of Biotechnology was established in the year 1997, and we started with only 5 students. And I'm very proud to say that now we have around more than 50 students per batch. That's approximately 250 candidates. So this department is actively engaged in cutting-edge research across multiple thrust areas to develop sustainable biotechnology solutions addressing global challenges. Our key focus areas include plant and animal biotechnology, where we work on genetic advancements to enhance agricultural productivity and animal health. We also work in the field of cancer and molecular biology, where we are investigating the molecular mechanisms of cancer to develop targeted therapies. Our research also includes phytomedicine, and cancer biology and explores the potential of plant-derived compounds. And we are also working in the field of making the functional foods. Additionally, we are advancing nanobiotechnology to develop innovative drug delivery systems and diagnostic tools. Our work in virology focuses on understanding viral pathogenesis and developing antiviral strategies. Through these multidisciplinary efforts, the department is committed to driving scientific innovations for a healthier and more sustainable future. So we have with us a team of experienced faculty members who are working day and night in different fields of biotechnology. Our commitment to sustainability is reinforced by the Center for Environment and Sustainable Development, led by Professor M.Z. Abedin, under the guidance and support of Honorable Vice Chancellor. The center plays a vital role in advancing research and innovative solutions for environmental conservation and sustainable development. To achieve the impactful research outcomes, we also collaborate with leading national and international institutions, industries, and government bodies. These partnerships help bridge the gap between the laboratory innovations and real-world applications, ensuring biotechnologies serve society in tangible ways. These departments were actually established with a vision to be recognized as a department of international repute with a strong interdisciplinary research. We also encourage entrepreneurial initiatives and have been actively supporting startups in biotechnology and bio-based industries. By nurturing young innovators, we are paving the way for future-ready solutions that align with India's vision of sustainable and self-reliant economy. Here I would first like to express my sincere gratitude to our founder of this university, late Abdul Hakim Saab, who dedicated his whole life to pursuit of excellence in education and community service. We stand here because of his contributions. The conference is an opportunity for all of us, scientists, policymakers, and industry experts, to engage in meaningful discussions and collaborations. The solutions we seek require multidisciplinary approaches, knowledge sharing, and technological innovations. As we embark on this 3-day journey of learning, let us collectively strive to develop biotechnological solutions that are not only scientifically sound, but also environmentally responsible and socially inclusive. I extend my sincere gratitude to all the organizers, speakers, and participants for making this event possible. I am confident that the discussions here will lead to transformative ideas and actionable strategies that contribute to a sustainable and resilient future. With these words, I welcome you all once again and encourage you actively participate, exchange your ideas, and collaborate for a better tomorrow. Thanks. Thank you.
Thank you, Dr. Mara Faruqi. Dr. Sanjay Kumar holds a master's degree from G.B. Pant University of Agriculture and Technology, Pantnagar, and a PhD from the Indian Agricultural Research Institute, New Delhi. He received postdoctoral training at Texas Tech University, USA, Rothamsted Research, UK, and Kansas State University, USA. Joined CSIR, Indian Institute of Himalayan Bioresource Technology, Palampur, Himachal Pradesh, as scientist in 1990 and superannuated as director of the same institute in 2023. Also during this period, held additional charge as director of CSIR Institute of Genomics and Integrative Biology, CSIR Central Scientific Instruments Organization, and head CSIR HR DG. Currently, he is chairman Agricultural Scientists Recruitment Board, New Delhi. His major contributions in research include discovery and transplantation of a novel carbon fixation pathway to minimize photorespiratory losses, leading to enhanced photosynthetic efficiency and yield, work lauded by international fraternity. discovery, characterization, and modification of an autoclavable superoxide dismutase from a high altitude plant, deciphering plant adaptation mechanisms at high altitude, mechanisms of prickle formation in rose, winter dormancy, and drought stress in tea, and understanding plant microbe interactions to portsum. His contributions on genome and transcriptome sequencing of Himalayan plants and microbes to decipher adaptive mechanisms and secondary metabolites synthesis are valuable. Dr. Kumar has guided 29 MSc PhD students, hold international patents, and more than 210 research review articles, book chapters, edited books. Key contributions have percolated to book and major reviews as well. He is fellow of INSA, NASI, NAS, Crop Improvement Society of India, CHAI Honored Fellow Award 2024 by the Confederation of Horticulture Associations of India. Bestowed with prestigious Vasvik Industrial Research Award, INSA Young Scientist Award, Certificate of Merit in CSIR Leadership Program, Ultra International Team Award, Lifetime Achievement Award by Mobilization, R.D. Asana Endowment Lecture Award, Professor G.V. Joshi Memorial Lecture Award, Professor Sri Ranjan Memorial Lecture Award, S.K. Talpatra Memorial Oration by D.U. Vasu Mathura, Dr. Subodh Kumar and Mamta Mukherjee Memorial Award by SFE, recipient of the Outstanding Alumnus Award from both G.B. Pant University of Agriculture and Technology for CBSH and the Indian Agriculture Research Institute. His new initiatives include, as director of CSIR IHBT, he vitalized several initiatives for empowering society, farmers, which include successful procurement and introduction of asafoetida, tulip production technology, monk fruit and Russian varieties of sea buckthorn in the country, introduction of saffron, cinnamon, licorice in Himachal Pradesh, lilium in Ladakh, and apple in northeastern region, technologies for waste management and vitamin D enriched shiitake mushroom production, combating malnutrition, preserving traditional knowledge, and conserving biodiversity. He also promoted aromatics in mountain farming system that positioned Himachal Pradesh as the top state in the country for producing essential oil of wild marigold. He coordinated multi-institutional projects of CSIR as Director of Agri-Nutri and Biotech team, as well as nutraceuticals and nutritionals missions. In the field of entrepreneurship development, he has put a strong emphasis on bioeconomy. The number of technologies and products developed by CSIR IHBT surged to 85 during the relatively short period of 2015 to 2023, that was his period as director. Several of these were commercialized to ensure their availability in the market, pioneered the establishment of startup culture, resulting in the launch of 56 ventures in the institute. With relentless approach for quality science and technology development, he led CSIR IHBT to attain top position in the state and among the top 10 institutes of CSIR as per the Shimago rating. With immense respect and admiration, I invite our esteemed chief guest to deliver the plenary address.
Very good morning to you all. Honorable Vice Chancellor of this great university, Professor Dr. M. Afshar Alam Sahab, Post Director Dr. Abdin Sahab, my good friend for the last more than 33 years, I think 38 years, Dr. Abdul. I think 38 years we are almost together. Professor Raisuddin Sahab, Professor Suhail Parvati, Madam Faruqi, all the dignitaries in this hall, young students, once again, very good morning. Indeed, a great pleasure to be in this great place. You will all agree, our summers do not pass unless we have to observe hunger. It's a household. And winters we can't survive unless you have Swalin with you. So Hamdard has been a household name. If I start naming these products, I think no household, at least in India, is complete without something. And unless you come here, then you realize you have a whole ocean and how such products are developed. I remember interacting with Dr. Abdin, going to his lab, and he used to tell me that these are the products which will be evaluated, and once they are evaluated, then they will be pushed into the market. I think this university, that way, is a classical example of value chain. Where very few universities you will find, if you think something and if you take it to the market, you have complete value chain of it. Sometimes staying within the university, one may not realize, but once you are out, then you realize it. Because that's what I had experienced, and I'll share some of the experiences, particularly when you try to take biotechnology to the market. I'll try to share. And when this theme, he told me that it is on global challenges, and then biotechnology has to give solution. I thought I'll try to see what I can contribute in this important area. I'm sure in this 3 days deliberation, when I was going through the content, I think so marvelous content. Top class people are coming here, who will be sharing their experiences with you all. And the audience, and particularly our youngsters, I think they have a lot to take, and their bag will be full, and then they need time to sort it out. But I'm sure each fruit will be so useful that some unique thing will come out of this. So I'll share some of my experiences in the past, and let me tell you, uh, I'll be sharing with you some of the work when I was at ISBT Palanpur. So I thank all our staff who were there at that time, and all our young scholars who are occupying now good position, what they have contributed, and they are occupying, some of them are in universities, some are in institutions, and I'm basically presenting their work. And certainly I was associated, so I was fortunate to be with them. Now, one thing I would like to mention with you all, that if you say that you want to do some innovation, first and foremost thing is important for you to know that what are the challenges. And if you know what are the challenges, then you achieve what you want to do. You might remember in early 1950s, we hardly had any food to eat. And today, we are food surplus nation. We are exporting the food. So we realized that problem. Our agricultural scientists, they worked hard. And today, we are exporting what 54 billion. I think it's not a small sum. It's a great, huge success done by all our scientists. So all breeding work, biotechnology work, it has gone into achieving this self-sustenance. My second view is that although today we say that we are self-sufficient, but what are the current challenges? We all are aware the way our population is going up. By 2050, I think we need to enhance our food production at least by 70%. And that too when your land is shrinking, that too when climate change irritations are there, you all are aware of that. And in the process, you all are aware that malnutrition is also creeping in. Although our bellies may be full, but do we have nutritious food to eat? And along with this, waste management poses a significant problem. And these are some of the areas where biotechnology really can play a very significant role. Even today, we are importing oil. Even today, we are importing pulses. And that's a great challenge. We are a feed deficient country. So our youngsters have to understand that whenever they take up a challenge, they should try to solve some problem which is of national importance. And that's where we need to focus. So this is one challenge. Now, my second issue is that whenever we try to solve any challenge, ultimately we also think of creating some economy in the process. And today you must be aware that there are several initiatives which are adding to the economy. For example, what I'm showing here is that industry, particularly which is based on bio-based. For example, you talk of biofuels, you talk of bioplastic. This is a very major industry. Enzymes, We are all importing in this country, but still some of us are manufacturing it. Around 70% we are importing. So this at least contribute to more than 48%. It is our biopharma section, and that is enriching our nation. Second is our bioindustrial section. It's contributing very significantly, and I have mentioned here bioagri, biotech, services. So these are four major areas in our nation which are contributing towards the economy. So we are aware that we have some challenges, and also there are processes which are adding to the economy. And if you see today, around 4.25% of our GDP is contributed by bioeconomy, and that is very significant. And therefore, our youngsters, they should take advantage of these new initiatives where we can move forward. And therefore, conferences like these biotechnology-based conferences, they are so important and they contribute so significantly. Just to give you a few examples, I have just listed a few companies, and you can see all of them are earning in dollars, millions and billions, and that is so significant. And we certainly have to look for that how much we can further add to this. Now I'll give you some examples what we have done in the past using biotechnology, what challenges we have taken, and what solutions we have given, and ultimately took it to the industry or to the stakeholders. Just to give you one example, we realize that we all are enzyme deficient country. All the enzymes in this country, approximately 70% come from outside. We said that can't we have at least one or two set examples where we have our own enzyme production system? And we thought that we should try to have the enzyme which has some uniqueness. Otherwise, you don't have patent, you don't have technology, how you go to the market. So for that, what we realized that we should try to target some areas which are very unique. So we tried to target an area which is a Kunzam Pass, which is at 15,000 feet. And we thought that since location is so hard, any plant or living organism surviving there will have certain mechanism to tolerate that sort of scenario. And we thought that why not we take advantage of that. So we focused on this, and when we worked out, what we realized that enzyme known as superoxide dismutase, which removes actually free radical, free oxy radical in our body, and they converts them into hydrogen peroxide and water. We said that why not we make use of this enzyme, because you know in all our, for example, aging process, you need this enzyme, right? You want to delay enzyme, you need to stop this enzyme, and it has lot of implications. So we targeted on this enzyme, and very interestingly, this enzyme you can autoclave and it still tolerates. There are very few such scenarios where protein can be stable upon autoclaving, and this was one such example. And very interestingly, this enzyme can function even at sub-zero temperature, minus 20 degrees Celsius. This enzyme is functioning. So we thought that this enzyme can have a lot of industrial application. So we cloned this enzyme. We modified this enzyme so that it can tolerate autoclaving at least 3 times. It should be, and also it should be stable at room temperature. So we engineered this enzyme. We did complete molecular biology. We did complete crystallography. So we had the complete knowledge of this enzyme with us. And once we were ready with this, we developed a production system. And in 3,000 liter bioreactor system, we developed the enzyme production, purification, everything. And when we saw that yield was around 350 milligram per liter, I think that was very significant higher yield. Then we took it to the industry. Industry at that time, around 15 years back, they had given us 1 crore rupees just to take this technology, and then they are putting it into the market. So this Phytobiotech, this company is a Kolkata-based company, and they are doing a lot of export business with this enzyme. They are doing pretty well. So this was one very successful example, at least right from discovery to going to the industry. Then also we thought that this enzyme can be used for plant improvement purpose, particularly under stress condition. When high temperature is there, most enzymes get denatured. At that time, this enzyme will remain stable and might impart tolerance to the plant. So we developed also transgenic, and we realized that this enzyme can give, it can make the vascular system very strong, so plant does not collapse under stress condition. So one enzyme at one hand it is going to the industry, on the other hand it is serving the agriculture purposes in terms of enhancing the stress tolerance. So this was one story which we just started right from discovery to the industry and helped in generation of economy. And I think in biotechnology, unless you go to the industry, unless you go to the stakeholders, what do we do with the biotechnology? So this one example I thought I'll share with you. Another example I'll share with you is that, in agriculture, everybody wants to convert C3 plant into C4 plant because people say that we do not want photorespiratory loss. Those who are dealing with plant, they should know that during photorespiration, carbon dioxide and ammonia is released. So we thought rather than minimizing emission, rather than stopping C3 and the photorespiratory process, which is very essential for plant survival, we thought, can there be some other mechanism or not? And to understand this, we again went to high altitude. And very interestingly, when we try to work out the mechanism, what we found that at high altitude, there the C3 plant, you take from low altitude to high altitude where there is reduction of CO2 partial pressure by at least 40%, still you don't find any change in photosynthesis rate. And then we try to work out the mechanism. We did the classical experiment of what people, Kelvin had done to trace the pathway of CO2 fixation. And what we found there, that those C3 plants take advantage of some C4 enzymes, and it's a basically hybrid of C3 and C4. And what plant does it? Whatever carbon dioxide and ammonia which is liberated during photorespiration, it refixes it. So we found it out. And there can be other ways of CO2 fixation, other ways of carbon and nitrogen fixation, which is emitted during photorespiratory process. So, and it was lauded, even if you see our F1000 prime and faculty opinion, they had actually picked up our paper and they said that it is a very, very significant publication. And rather than converting C3 into C4, this is a very useful path. And if you see this book, Plants in Alpine Region, they have really quoted us so extensively and several chapters just based on our research. So what my point to the youngsters is that I think if they try to go to some unique locations and try to see the survival of living organism there, certainly some new things can be there. And they can add a lot to the knowledge. Not necessarily everything has to come from the West. I think you guys are here. You have to contribute very significantly for enhancement of knowledge in whatever field you want. But you need to identify the right niches. That's very important. And one slide I will know, because I know Dr. Abdin Saab is expert in artemisia and secondary metabolism and metabolic engineering. And I remember his true love for artemisia. Am I right, Dr. Abdin Saab? Your love for artemisia, apart from other plants, and the nanotechnology he uses. So I just for you, I put one slide because we had done some work on secondary metabolism on a range of plants, particularly on high altitude plant. And our whole idea was, can we transplant this pathway into some other system? Initially, we succeeded in tobacco, tea production in tobacco. But it was just for the scientific curiosity and scientific interest. Nothing to take it to the industry. We don't want people to have tobacco in the name of tea, right? Just to show that. But anyway, we laid the basis of enzyme engineering actually through the secondary metabolic pathways. And if you see our publications in secondary metabolism, in most of the secondary metabolite of high altitude, you will find very significant information there. I'll not go into the detail. But what I will try to add something interesting is that, I mentioned about this waste management is a major problem, and particularly our plastic waste. And we need to identify the right sources how we can make bioplastic, for example. Now we have a range of things, but what very important. We had one micro, and this micro accumulates a lot of primary product for bioplastic formation. We recently got this patent just this year. In 20, we had filed this patent, but it took 4 years before we could get it. And one company which is incubated in our institute, they are already making this, and they are doing lot many innovation with this bioplastic thing. And here advantage is, sometimes if you want to make bioplastic, economy is very important. You want one thela which should not cost more than 10 paisa. If you say that my thela is costing 1 rupee, nobody will buy it, right? आपको तो सब्जी खरीदने, you want it free. That थैला का आप पैसा थोड़ी देना चाहते हैं. So the cost is very important. So to ensure that, we took out one bacteria where bioplastic is a byproduct and major product is a pigment known as violacin. And violacin takes care of all the financial aspect, and this helps the environmental aspect. And just because of this reason, bioplastic was not successful, but now since we have a micro which can offer you this advantage, so this company has taken it, and they are now supplying and making this bioplastic because they have a pigment formation from there. They are making a lot of money, and bioplastic they are using it for the purpose of making all your bags and other stuff. So this is one unique thing. And again, this bacteria we got from very altitude, from very stress environment. So again and again, what I am stressing to particularly our youngsters is that they should always look for some novel niches. If they look for some novel niches, they are going to get something very novel and very relevant. Now, another thing, next, so these were some of the examples I wanted to share, that we had some innovation and we took it up to the market level. Next, I want to focus is that whenever we do any such things, value chain is very important. Sometimes we say that we want to push XYZ product into the market, but do we have the value chain or not? And I'll give you some examples that how value chain creation helps us to link lab, farmer to the stakeholders and ultimately help them to generate some economy. See, in my talk, let me tell you, whatever work we do, I always say that unless the money comes into the pocket of the stakeholders, I think whatever we do in the lab, we all are sustaining ourselves on taxpayers' money. We owe something back to the taxpayer. They expect from us that we give back something to them, right? So therefore, throughout my life, I try to do something to give back to the taxpayer, that we have taken some money from you and we will try to give it. Right? So that spirit, I think if we all develop, everybody will respect a lot of for our scientists. I gave you one simple example of from the right from blue of the to solid, but there are several such products, right? If those products are not there, do you think Hamdard will have its that name which is in everybody's name today, right? So that spirit, I think our youngsters should imbibe it, and they are here at the right place. So my example here, I am giving you one aroma mission. You all will be surprised. In India, even today, all essential oils are imported. At least 40 tons of rose oil is imported in this country. We don't even have rose oil. Wild marigold oil, all come from South Africa. Bulgaria gives us rose oil. From old time, we are known to produce it. From where now? When we don't have the oil, essential oil, what do we do? Essential means any oil which gives you nice aroma. So aromatic oil, we realized that it is a great potential, right? So we started with very simple thing. We said that if we have to ensure that our country makes a lot of aroma oil, we should have the value chain. And value chain means you should have the variety. Here the role of breeder and biotechnologists come. You should have nurseries so that planting material is not limited. You should have cultivation practices because farmers should know how to grow it. You should have a distillation unit because if farmer produce the flower and if he sells in the market, it will be selling 10 rupees a kg. If you say that you extract oil, it will be 6,000 liters, right? So that advantage we want to give to the farmer. So we have to make technologies where the farmer can use it. Then extraction process, we have to tell to the farmer. Quality assurance, we have to teach to the farmer. And value addition, if any possible. Then storage, very important, and then marketing. If you have these 9 points, then only you can achieve something sustainable. So we took it as a mission mode, this project, and in 2 years, we were importing around 12 tons of marigold oil from South Africa, and just in Himachal Pradesh, 8-ton oil people started producing. And from the land from where they were not getting any money, everybody was getting at least 1.25 lakh rupees per hectare. And that was very significant. From rice, you get at least 40,000 rupees, right? But here again, whether it was a biotechnology, whether it is a breeder, whether it was a chemical technology, everybody came together, handed over the technology to the farmer. Initially, we spent around 20 crores, and farmer got 100 crores. I think our return on investment was 5 times. And then what else one needs? And I think that's how we proved one case. Another, then we developed a lot of startups around it. Today, everybody talks of a startup, but one problem, good people don't realize it, and we solved that problem. You see, when people come for doing PhD, everybody gives them fellowships. But anybody comes for a startup, they say you spend your own money, right? Is it logical? It's not logical. We said when we can give money to the people who come for doing PhD, why can't we give money for the persons who want to set up their own startup? So we started giving them 25,000 rupees per month for 1 year, but it was a conditional. And I think if you put that conditionally condition into PhD guys, we said if you don't start your startup after 1 year, you'll have to return all the money. It was a conditional fellowship. State government agreed. And then also, once a startup made the product and wanted to put it into the market, they wanted money. And when they went to the bank said you have to give collateral. Collateral means girvi rakhna. You have to give collateral. Now person said, "Are mere paas ek ghar hai. If I put that into girvi, and so tomorrow my product is lost, I'll be nowhere." Correct? So we signed an MOU with SBI. We said that anybody who takes our technology, wants to put it into the market up to 1 crore rupees, he said we will not charge any collateral. And I think that was very significant. And that was, since it was from our side, so they agreed for that. And that enabled a startup environment. And so many startup, I'll give you at the last, so many startup came up only just by this small change, and government agreed. And you see how many people started associating with us as we started moving into this aroma range of product. And now I think even after my coming back, not many people started. So this was a very successful is flowers. Everybody loves flower. You see, your whole this stage is full of flower. No conference without flower you will ever see. But have you ever thought from where those flowers come? And we thought, why don't we focus on some of these nice flowers? And you all youngsters will be surprised. All tulip come from Netherlands. All lilium come from Netherlands. We don't even grow our own tulip for Netherlands. Just to give you some example. All peonies, they all come from outside the country. We don't have our own peonies, and that is a scenario. So we focus on some of these flowers. And one of our success was this tulip production in India. You must have seen Srinagar Tulip Garden. All bulbs are produced from Netherlands. 15 crore rupees bulbs, they come from Netherlands and they show it to the public. I said, yeah, what is this joke? I said, our farmers should have the capability to grow their own tulip. So all these, what you see this garden, all bulbs were produced by our own farmers. So here tissue culture, biotechnology was again very important. So we had that technology, gave it to the farmer, and we established the first tulip garden in the country, which was by the bulb which was produced indigenously. That was our first contribution. And it was not limited to that. When this Ram Pratishtha, Ram Mandir was there, no? Our tulip went from Palampur, and it made the news headline that at least all the flowers which were given, it was made in India, at least something significant. And very important, one more thing. Very recently you must have read in the newspaper, Delhi has lot of tulip. And before I came, Delhi, Honorable LG, he was member of our research council. At that time, he was not the LG. At that time, he was chairman of the KVIC. So he was member of our research council, and I used to discuss with him that we are working on this tulip. So when he took over as LG, first thing he said, and I used to tell him that in Delhi, in Rashtrapati Bhavan, you put all the tulips which are getting from Netherlands. Why don't you give us those bulbs? We make it, we produce it, and we'll give it back to you. And you have all the bulbs from India. And this time you read in the newspaper, it says that at least 15,000 bulbs which were produced indigenously. And that was from ISBT Palampur. That this is the Hindustan Times. So these things give us satisfaction. So what my point is, whatever you do in the lab, it should always percolate down, like a रूह है. Suppose रूह अफ़ज़ा आपके हमदर्द में रहता तो हमारी रूह कैसे तृप्त होती, right? So something of that nature has to be done, and without that, science cannot develop that sort of respect. Another, I'll give you an example. People say that Ladakh is all sandy area, nothing happens there. We said Ladakh is a gold mine. Suppose you want to produce lilium, you want to produce tulip, that's the place. Once we went initially to Ladakh, we never saw any flower in that area. And now we gave them this technology of lilium bulb production. And today you see, whole Ladakh, our Leh area, Kargil area, it is full of lilium production. And today there are several shops. And namaskar, sir. Namaskar. So now you can see first shop was opened, and we inaugurated that shop. First flower shop in Leh was opened by us. And now this all flowers, they for the first by first flight it comes to Delhi. Now again, lot of biotechnology like our tissue culture technology and all those things played very significant role. But ultimately we gave it to the farmers. So right from tulip to lilium, now you can see the empowerment of the farmer, that every morning it is their job to send all the lilium bulb to Delhi market. So that was yet another significant achievement. And certainly it was backed up by high-end technology like hydroponics, aeroponics, and several companies have taken this technology. So all this off-season tulip production, it was all through hydroponics. And it sells a higher cost. So that return on investment is always there. That we always calculate. If we spend 10 rupees, at least farmer should get 15 rupees also. So these high-end modern technologies are certainly part of that. And you can see how many people started associating with our institute at that time. So on again floriculture base, we developed a lot of products and tulip. Another one thing I wanted to mention is on the honey production. Right, in honey production, we are even in 7th position, and we wanted to be somewhere near to China. And one of the reasons what we realized that in honey, our honey hives are very classical honey hives. People had to squeeze honey hives and they kill a lot of larvae. So we had one institution in CSIR at Chandigarh, CSIR. Along with that, we had developed a honey hive where you don't have to really squeeze honey, and you don't kill any larva. So they had developed very nice honey hives for us. And using this honey hive, we developed several startups. And without killing any larva and honeybee, you can extract the honey from that honey hive. And it was a very successful stuff. And now we had developed one honey garden, because in honey production the major problem is that during off season when there are no flowers, you have to transport the honey boxes. So if you have a honey garden, you can design it. Yes. So that would be very useful and significant. And whenever we try to transfer some of these technologies, we use our mechanism. For example, MSME has very good scheme of Spooty. And using this Spooty scheme, we have transferred and percolated and diffused several of lab technology into the villages itself. So they have the production center in the village, and that's how we try to take it. Now, so these were some of the examples of the value chain creation, that how we created value chain, empowered our farmers, and everything went into the market. Another thing, and now I'll give you some of the stuff on which we thought that There are several even agricultural commodities on which India is dependent on export. And one such example was hing or asafoetida, right? And every household consumes it. And we realize that we are getting all hing either from Afghanistan or Iran or Uzbekistan. So we first of all we introduced this hing into this country, and what we also did is, once we got these hing seeds, our problem was their germination rate of almost 1%, almost negligible. So we developed technology, and today we have the 90% germination rate technology. And once we developed all this agro technology, we signed up agreement with our state government. The state government supported our activity. We linked up with the KVKs, Krishi Vigyan Kendras, so that whatever we develop in the lab, it goes to the farmer's field. And today you can see we have around 250 sites where hing seed is being produced, and around there are 93 villages which are cultivating this. And this year we expect first hing to be coming out of this plant. So this year we are expecting, and I'm sure that first indigenous hing made in India will be there. And once these things are there, now we will take it also to Uttarakhand, because Uttarakhand is yet another area, because our cold desert area is the right site for the hing production. So this has been a very successful thing. And just on the lighter side, once we introduce this hing, I think right from UK to USA, apart from India, everybody covered it extensively. That India, who consumes so much hing, at least now started producing hing itself. And then when in Kaun Banega Crorepati, this question came, that was again, he mentioned very nicely that at least there is a lab in Palampur which introduced hing for the first time into this country. So I think it has gone to public, it has gone to in everybody's mind that hing has come into this country. Yet another important crop which I thought I should mention is about this saffron. In India, it's mostly in Tempo region. Approximately 3,000 hectares is there where this produced. Approximately 9 ton we produce annually, and our requirement is 100 tons. And people said that saffron cannot be grown beyond Kashmir. We said, why not? We did a lot of ecological niche modeling, what you see here. And after that modeling, we realized that there are several areas where it could be produced. So we did some trial experiment, and we realized that Himachal Pradesh can be very nice area. Again, Uttarakhand is a very potent area. So at least in Himachal Pradesh, using our tissue culture comp, we developed this tissue culture technology. And also, there is a limitation, this saffron cannot be productive unless the bulb size is more than 8 gram or more. So we overcome that challenge also. And after overcoming that challenge, the saffron production you can see, now in Himachal Pradesh, we have 20 different blocks where the saffron is being cultivated. And now it is going more and more and more. So after him, the saffron was yet another example, very successful example that we did in Himachal Pradesh. And again, lot of tissue culture. I realize that tissue culture is such a powerful technology. At times we never used it, but this is a very powerful way when we have to multiply the plants in large number, and it was very successful. Another example I'll quickly rush through is that on apple cultivation in Northeast. In Northeast, you see most all their apple either come from Myanmar or they get from Kashmir. And by the time they get from Kashmir, it's already so much old. So they wanted some fresh apples. And their people are engaged in opium cultivation, which is not desirable. Once we got into this apple cultivation in Northeast, we signed agreement with the Northeast Agency. We empowered 5 tissue culture companies to make the root stocks. We made the cuttings ready, and right variety again. And they are very classical variety, which can give apple at low. Normally you require more than 1,200 chilling hours for apple. These variety needs hardly 500 hours of chilling, and even less than that. And they were very successful. And once we did initial trial, once it was successful, now you can see apple production in Northeastern region. Honorable CM there, he was so happy. He gave 1 lakh rupees to each farmer. They are bringing the state right from opium cultivation to this apple cultivation. And Honorable Prime Minister also mentioned in his Mann Ki Baat that at least Himachal Pradesh and Northeast, East-West connection is there in terms of apple cultivation. So that was something very encouraging for our scientists who were doing. Now it is supported by DBT also. Our this activity is going pretty well. And like this, we have taken several initiatives. Another initiative, a new crop we introduced in India is the monk fruit. Normally it has a molecule known as mongosite, which is 300 sweeter than the sugar, and it does not have any aftertaste like stevia. It's grown in China, processed in USA, and supplied throughout the world. And here we did it for the first time. We tried in Himachal. We did the complete biology of this plant. Again, tissue culture. We have multiplied the plant system with the state agency. We evaluated in several areas, and now it is ready to expand within the nation. So, like this, I'll just quickly go through that 4, 5 another crop we did. One is cinnamon. You will be surprised. All 90% of cinnamon comes from foreign countries. Although we say that Kerala is the center for cinnamon production, but there are hardly 200 hectares under cinnamon cultivation. And what we are growing in northeastern and other part of the country is the incorrect variety of cinnamon, which is known as cassia, and that has very high coumarin content, which damages our liver as well as kidneys. So we wanted that people should grow the right variety of cinnamon. So we introduced in Himachal Pradesh, and now at least 1 lakh plantation we already achieved. But again, a lot of multiplication, agrotechnology, that work went in at the background. And another crop that we did is on the licorice. I remember, sir, I think in CMAP at one time was trying to do something on licorice. But yes, but at least in Himachal Pradesh, we now introduced very successfully this licorice, because again, all licorice come from Afghanistan. And I think HOU has the only some exceptions they had HAU, then CMAP was doing something, but in Himachal we introduced this licorice. Mulati, you see, in India we don't grow even mulati, we don't even cultivate mulati. Another one thing, because youngsters are there, I just want to share one thing, that whenever you try to develop any project, it's important that the industry meet is very important. And we had one nutraceutical mission where we wanted to push some nutraceutical product into the market, and that's how we did some work on nutraceutical mission, and we took several such products into the market. It was around vitamin D, it was around for zinc and iron nutrition. So several such products were pushed into the market, and again we took help of MSME to push several of these products into the market, and we were attached with the malnutrition program or Poshan Abhiyan of Government of India, and that's how we pushed several of these products into the market. So it was again a value chain creation. Our entrepreneurs were producing, and we had a connect with the state government in terms of pushing some of these products. And one thing I wanted to mention, that always use the existing mechanism of the government in terms of technology transfer. And here I am giving you example of this MSME Spurti scheme. We made a lot of use of these schemes, and we established several centers in the villages to take some of these products into the market. And today you see the processes that I told you. Using that process, we had developed 56 startups, and their all products are into the market. And we developed around several technologies, and around 40 technologies were given to around 83 entrepreneurs. And you can see one technology to multiple entrepreneurs we had given. And I'll just show you some of the lists, that these are so many technologies we pushed into the market, and it all were very, very successful and developing a lot of and adding to the bioeconomy to this nation. And in the last time, I will just mention that I think it's very important that you identify the right challenge. Then only you can be creative. Then only you will be problem solver. Sometimes risk has to be taken. You need to take that risk. And value chain, it has to be created. Unless you have a value chain creation, you can never ever develop that respect and benefit the society. So with this, I thank you so much for your patience listening, and I'm sure that this conference, Dr. Abdin Saab, will take you to a very new dimension. I'm sure also a speaker like Dr. Khanuja Saab is here in the audience. And actually, when we all were students, we always used to look at him. He's always full of new thoughts, novel thoughts, innovative thoughts. I do not know what I can say. And not only thoughts, he would convert those thoughts into action. And that was important. So whatever we learned, so happy Dr. that you are here and I had a chance to speak something before you. And thank you so much and all the very best for this conference. Thank you so much.
Thank you very much, sir. Your thought-provoking address set a perfect platform for our speakers to deliver their presentations. Our Honorable Vice Chancellor, Professor M. Afshar Alam, sir, apart from being a noted academician with a strong administrative acumen, is our torch-bearer with strong leadership and vision. Integrity, excellence, accountability, transparency, and teamwork. He has created a great workplace atmosphere to carry everybody along in the organization to work tirelessly towards a common goal, and that is accomplishment of overall excellence. Very humbly and respectfully, I request our Honorable Vice Chancellor, sir, for the presidential remarks.
Thank you. Good afternoon to all. Dr. Sanjay Kumar, the chairperson of Agriculture Scientists Recruitment Board, Ministry of Agriculture and Farmers Welfare, Government of India. Professor S. Raisuddin Saab, the Dean Academics, Professor Sohail Parvez, the Dean of the School of Science and Chemical Sciences, Professor M.Z. Abdin Saab, the Organizing Secretary of this international conference, Dr. Humaira Farooqi, the HOD, Dr. Saima Wajid, the convener of this conference. I'm very happy and glad that National Conference cum workshop on Sustainable Biotech Solution for Global Challenges has been organized by the Department of Biotechnology in collaboration with the Department of Science and Technology and Department of Biotechnology, Government of India from February 19 to 21st, 25. I'm sure this gathering brings together some of the finest minds in biotechnology, united by the shared commitment to advancing sustainable solutions for global challenges. We are pleased to have Dr. Sanjay Kumar, the chairperson of Agriculture Scientist Recruitment Board, the Ministry of Agriculture and Farmers Welfare, and as our chief guest. And his work in agriculture research and biotechnology has been instrumental in promoting sustainable agriculture practice, ensuring food security, and addressing the global challenges, especially in floriculture and tissue for the plant, because he has already presented a wonderful presentation and which he mentioned how his Palampur Scientific Center is working for that. Sir, we had already done a lot of last 3 years back for honey breeding and some kind of floriculture in our campus also. And we had also planted some of the apple tree. And last year I took tree, but that was not very, a good apple, but it was Delhi apple. Yeah. So biotechnology play a transformative role in addressing some of the most pressing challenges in our time, from food security and climate change mitigation to healthcare advancement and environmental conservation. Of biotechnological innovations are shaping more sustainable future. The world is facing multiple crises, that is climate change, resource depletion, biodiversity loss, and global food insecurity. The need for sustainable solution has never been a greater. Biotechnology offers innovative approach to tackle this challenge through a green technology, bio-based solutions, and a circular bioeconomically that balance economic growth with environmental responsibility. With the growing global populations, ensuring food security while maintaining the ecological balance is crucial. The biotechnological advancement in agriculture offers solutions such as climate resilient and highly yielding crops that can withstand drought, pest, and disease, biofertilizer, biopesticides that reduce chemical use and enhance the soil health, and precision agriculture using the biotechnology and artificial intelligence for the efficient resource utilization. Those advancement biotechnology is driving a shift towards a sustainable agriculture practice that support both farmers and the environment. Advances in biotechnology are also revolutionizing the healthcare, biopharmaceutical, gene therapy, and synthetic biology that are transforming medical treatment, making them more effective and personalized. These breakthroughs are enhancing healthcare outcome and improving the quality of life for people worldwide. A circular of bioeconomically is essential for a long-term sustainability. By leveraging biotechnological innovations, we can convert agriculture and industrial waste into a high-value product, promote renewable bio-based materials for sustainable manufacturing, and support of zero waste and low carbon production model for a healthier planet. With the biotechnology in its core, the bioeconomy is set to become a driver of the economic growth, creating new opportunities in a green industry. Biotechnology offers powerful solutions to mitigate the climate change and the environmental degradation by driving innovation that promotes sustainability. The development of biofuel and bioplastic is reducing dependency on fossil fuel and minimizing the plastic pollution and offering eco-friendly alternative for energy and materials. The bioremediation technique has been employed to clean up industrial waste, oil spills, and other environmental pollutants, restoring ecosystems and ensuring the cleaner the natural resource. Additionally, a carbon sequestration technologies are playing a crucial role in reducing greenhouse gas emissions, helping combat global warming. Collectively, these advancements are facilitating the transitions to a greener and more sustainable economy while significantly lowering the environmental footprint of industrial and agriculture activities. At Jamia Hamdard, sustainability is an integral part of our academic and research framework. Our effort towards a plastic-free campus, eco-friendly research practice, interdisciplinary sustainability initiative reflect our commitment to the environmental conservation and responsible scientific progress. This conference being together the scientists, researcher, industry experts, and policymaker to exchange the ideas, share innovations, and build collaborations. I appreciate Professor Amjad Abdin, the organizing secretary, for his dedicated effort in this conceptualizing and organizing this important event. His contributions to research and academic excellence have strengthened the Department of Biotechnology and created a dynamic scientific environment at Jamia Hamdard. To all the participants, this is an opportunity to engage in a meaningful discussion that will shape the future of biotechnology, and the insights and collaboration formed here will have lasting impact on research, industry, and society. With this thought, I again welcome you to all the participants and look forward to a productive and insightful conference. Thank you. Jai Hind.
Thank you so much, sir. May I now request Professor S. Raisuddin, sir, to deliver the vote of thanks.
Assalamu alaikum and a very good afternoon. This is my duty to present vote of thanks for today's program. At the outset, I like to congratulate the organizing committee under the leadership of Professor Amjad Abdi, who was head of department for a considerable point of time of this very, very important department in our university, which has flourished, and we have UG, PG, PhD programs very much demanded. So, organizing committee and also our Honorable Vice Chancellor, who has been promoting very much research and academic culture on the campus, encouraging the faculty members, head of department, deans to have collaboration, to invite distinguished persons on the campus to conduct the programs like this. And every week, every month, I think we have a very, very important conference going on the campus with the support and encouragement from our Honorable Vice Chancellor. At the dais, we have a very, very important person, Dr. Kumar, who has spoken a lot about how the small ideas which can be good for the country can be translated into the market. And from discovery to market, his emphasis was. And there are many vehicles now. He mentioned about Spurti, for example, MSC, small and medium industry to enterprise. I think from there, grant can be obtained. And we have now this kind of opportunity in our country where government and government divisions and departments are promoting startups, taking small project to the market, to the industry, commercialization. So long back, that kind of ecosystem was not available in our country. But now, with this government in particular, they are asking that make in India and produce in India certain programs. So I think the delegates who have come here can be inspired. I'm very much inspired that he mentioned about 10 to 12 products starting from the enzymes, from the floriculture, from the heme, and also about the saffron and so many other products and how they can be, they were successfully grown in certain areas where it was not earlier were grown. So thank you very much for sir, very much delightful. I'm so impressed with your lecture. I would also like to thank on the dais, we have our young dean, Professor Sohail Parvez. Under his leadership, this school is making a lot of progress and a lot of academic and research activities are being inculcated and enriched in the school. I also like to thank young head of the department, Dr. Humaira Faruqi, under whose leadership now this department, I'm sure, will flourish as Professor Abdin has laid a very strong foundation. So now she will take forward these initiatives. Dr. Professor Abdin is already there under his mentorship. I am sure that she will do a lot of such good activity. Also, other members from the school, Dr. Saima Wagis, who was compared at this program, Dr. Monica Safi, Assistant Professor, Dr. Jagriti Naran, Dr. Sheikh, Javed Sheikh, and Dr. Javed, and we have Monica Safi. These are the young faculty members of the department and have contributed a lot to the successful planning of this program. So I also like to thank all the deans, head of department, faculty members, and students, and welcome all the delegates. And thank you for coming to the university for attending this program for 3 days, which have element of conference as well as element of workshop. So I think they will visit to the department, the facilities in the field. We have a very good herbal garden. About 6 acres of land is devoted for the herbal garden. So more than 80, I may be corrected by Dr. Iqbal Azhar. How much varieties are there? Huh? More than 100 varieties of herbal garden. So very eminent person like Dr. Kanuja is there from CMAC. He was director, I guess, for a very long time. And we are lucky that our first vice chancellor was from CMAC, Professor Akshay Hussain Sahab. So, and also I like to thank Dr. Toteja, G.S. Toteja. He was the additional Director General of Indian Council of Medical Research. Now he is associated with our university as adjunct professor in the Department of Biochemistry and Department of Toxicology. So I thank you for coming to the program, and I hope that, and many other people are there from other the institutions, and as a chairperson, as a speaker, as a plenary speaker, as invited speakers, I think these 3 days of this academic and research deliberations will be highly enriching for all the delegates who have come. I also like to thank all the volunteers who are contributing, And in the end, I like to thank the funding agencies, which is the important one, like Anusandhan Research Foundation, which was earlier SERB of the Department of Science and Technology, and we have support from Department of Biotechnology. We have also support from our own internal resources, so from Jamia, because we provide grant for organizing some conferences, and our Vice Chancellor provides very much handsome grant to the organizing committee. Besides, this conference is also supported by industrial partners like NextGen, Takara, Edur, TVS, Microlit, and many other small industries and which have come forward to support this conference on the sustainable biotech solutions for global challenges. So, and also like to thank the PR team, public relations team, and hospitality team, Dr. from our Sumer Akram Sahab, and Dr. Iqbal Aja is also very closely contributing this, will contribute for next 3 days to the success of this program. I also like to thank the registrar who has just left because of some important assignment he has in hand, but his role is also very important for getting approvals, for getting facilitation for this hall, for auditorium, for the guest house, and for the other transportation arrangement. I think he is quite, and he is one of he was earlier at very reputed institutions like Manu, and he was chairman of the book trust. So in that way, he knows about the academics as well as administration. So his role is also very important. I also like to thank the staff of the registrar office, vice chancellor office, and all others, the hospitality manager, Mr. Tanveer, and all. They have made very good program. So we hope that from Jamia Hamdard, and we have Jamia Hamdard, I hope that all the delegates will have very good enriching experience for 3 days. And please visit our facilities, Herbal Garden and the Department of Biotechnology, their greenhouse, and how many technologies they have transferred. One of the important techniques Professor mentioned, our chief guest, about the this Artemisia. So Artemisia technology has been transferred to IPCA laboratories, which is with the vision of our head of the Department of Biotechnology. And many more technologies are under process of transfer to the industry. So I hope that this will conference will be an enriching experience. So thank you very much. And please enjoy this.
Thank you, Professor Raisuddin, Dean Academics Director, IQAC. May I request all to please rise in your seats for the national anthem.
पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जय गण मंगल दायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
I invite all present for high tea served outside this hall. Thank you.
CSIR as a director that time of CMAP. CMAP is a very wonderful institution now, so our Central Institute of Medicine and Aeronautics Plants in Lucknow. And on his own merit basis, his outstanding publications and the wonderful research which he was able to carry out all through his career, he has been elected as a fellow of few of the academies, important academies of science in India. For example, he is a fellow of the National Academy of Sciences, he is a fellow of the National Academy of Agricultural Sciences. Professor Kanuja basically is a technology mentor to both agro and biotech industry in India. He initiated the Entrepreneur Enabler Platform. Please listen to me carefully. This is what we all must contribute beyond your capacity as a researcher and scientist. Professor Kanuja initiated this Entrepreneur Enabler Platform named as Suman Kanuja Innovation Enterprises, which is abbreviated as SCLS, in 2011 for technological innovations for novel bioagricultural product developments. He has shaped this green project, particularly the societal empowerment, linking both agriculture and industry with health sector. And the focus is on societal empowerment. And founded by him in 2015, he initiated, like a wonderful science foundation. We call it as Flora Fauna Science Foundation. Flora, fauna, I'm sure you all know it. We all belong to either flora and fauna, anything or everything on this planet Earth. So he initiated this foundation in 2015 and together raised experts and volunteers from different spheres of science, not only science, also business, farming, media, society, and academy, all together in one platform. And Professor Kanuja also mentored Helix Biotech Limited during 2009 and 12, and as a R&D driven industry with novel products in functional foods. Importantly, as a scientist, we must all applaud. He has developed 50 technologies and product patents and 173 research papers, publications in the field. This is the big number. He has won several awards of distinction, and few of them I may like to mention here. That he has won CSIR Technology Award in Life Sciences. He's won Golden Peacock Award, he's won Boswick Award, which is a very famous award in biosciences, and also Nina Excellence in Technology Award by IIT, Kharagpur, and many other awards he has won. So with this brief introduction, though I can talk about him a lot, and by the way, I must also recall here at this moment, I have a pleasure after a long time actually meeting him and had this pleasure, special pleasure to introduce to him. I would just like to recall my own initial introduction with him. When I joined, I hope he also remembers and recalls, when I joined as a PhD student in 1984, he was already a well-known personage student by himself that time in the area of cloning, genetic engineering. Big company in DNA was just taking birth. Of course, it was taking birth long time ago, but in India, that was a big name. Other than me, nobody knew. So I was supposed to give a seminar, in our PhD on genetic engineering, and I accepted that challenge, and I did not know anything. The first person I went to understand what it's all about was Dr. Kanuja at that time. And so I recall that, and I remember, with great pride that yes, we continue interacting with each other even now, and I'm sure you all will be happy to listen to his talk, which will be innovative talk, not a routine talk, which you will be expecting from a person like me. So with this, again, I have pleasure in now introducing to you all of you and to welcoming Professor Kanuja. Over to you, Professor.
Thank you, Professor Bansal. A very good afternoon to all of you. बोल तो दो भाई, afternoon. We, unless we smile, things don't go good. And Flora Fauna Science Foundation, which he mentioned, which is my child, not 10-year-old. This time, every year I have a theme for it, and this year the theme was Vigyan Se Muskan. So, thanks to smile. So, smile, how does it come? Is something that remains a mystery. People do not know why did they smile sometimes. but it is internal happiness. You cannot smile artificially, you will be caught. So that's what aim of my mission is. I thank the organizers of having invited me and then insisting Abdin and Javed and Monica that I have to come. So I came from Lucknow only for this purpose this time. Although I live in Delhi also, but just now we had our annual summit there. I call it Innovation Summit or Flora Fauna Innovation Summit. So I was occupied there. But then a sort of affection for this university. I was adjunct professor in this university between 97 and 2000. Dr. P.S. Srivastav used to be here, so we used to interact and talk about biotechnology at that time. So it's a bondage that I have, and Abdin being from IARI again becomes another bondage. It's good that I'm here for me. Okay, you will judge whether it is good for you of my being here or not. So when Dr. Abdin asked me and told the title of this workshop come conference to give a talk, I chose this title, which you are seeing here, Biotechnology and Next Generation Agriculture for Future Food Innovations. Well, I had been at IARI. I studied here in genetics division, and therefore my introduction is I'm a student of genetics, but a lover of food and nutrition. So I did stay here for 14 years. Then I moved to CMAP and moved from microbial systems or microplant interaction system to plants producing secondary metabolites. And then from there, I took voluntary retirement in 2008 to make a record that if any director of any organization can break that record, because I took voluntary retirement at the age of 49. So I always challenge CSIR directors, can you break that record? You might break the record that when I joined as director at the age of 42, but 49, you retire is difficult. The reason for that was that I wanted to translate science. And translating science is something very different than doing science. And doing science is very different than practicing science. So I did all of them, and I wanted to see both sides of the table. As a client, when people used to come to me, and I used to insist that in medicinal and aromatic plants, if you are working, you must become entrepreneurs. And they used to tell what are the difficulties of entrepreneurs. And I wanted to go and see that what are those difficulties and how can then I remove some of those bottlenecks, provide solutions. So that's how this title came. And this one? No. Page down? Okay. So with this title, then I thought, what could be, what I can convey as a message. I'm particularly focusing on students and young generation, young researchers who are at a decision-making stage whether to search job or provide jobs. And can we be entrepreneurs by doing science, not by doing only money making? I believe entrepreneurship is not profit making. Entrepreneurship is really translating something. So usually, I do talk about it, that what is science? Science is answer to the questions like what and why. What is it? If you answer it a very fine way, then it is science. Why is it happening? Then also it is science. And when we come to how is it happening, it becomes technology. That's why we call it techno-how. How? So answer to how is technology. And then, in fact, in technologies, it's when we move from know how to show how, and show how to do how. These three elements are very, very important when we want to translate it. But then there are questions which are opposite to it. And first question that is important is why not? My teacher says this happens like that, and I say, why not this way? And he says, ऐसे ही होता है भाई. So then, no, that's not the answer. I'll frustrate that teacher. So you should also do. All your teachers, those who say, ये भी नहीं पता, say yes, ये भी नहीं पता. But get the answer. And then say, आसमान नीला क्यों? पानी गीला गीला क्यों? So those things when you will start saying these are why nots. और फिर वो जब आप उस पे आ जाते हो, when you have come to that why not, then the answer will come from how not. कैसे नहीं होगा? So why not, when you start answering, you become innovator. From science to technology to innovation. And how not, when you do, you are an entrepreneur. So that's what my dream is, that the young generation should go towards that. And that's why after 49 years of page spent there with 1.5 tenure of director, I decided to become why not and how not man. And tell you that it is possible. Of course, there is a risk. You have to cover that. But the biggest risk in life is not to take the risk. Remember that. If you have never taken any risk in life, then you have taken the biggest risk. And therefore, don't be afraid of that. So, what I would like to define as food, because when we say agriculture, of course, there are many things agriculture gives, but one of the prime things is food. Making health sense in foods. Can foods be preventive healthcare systems? Can we even eliminate the use of medicinal plants by food? Or can medicinal plants be the food itself? Hippocrates said, let food be your medicine. But remember, Hippocrates did not say, let your food taste like medicine. That doesn't have to happen. That's where innovation will come. That you have to do organoleptic research and make food tasty as well as healthy. बच्चों को ये कहोगे पिज्जा नहीं खाना है, wrong thing. Make pizza healthy. Kids have to eat everything. They must eat chocolate. They must eat pizza. They must eat pasta. They must eat fast food. There is no junk food. Junk food itself is a wrong definition. Something can be either junk or food. How can you call food as junk? It is we who are making it junk. So we have to do research as scientists, as entrepreneurs, as students. We can think of. So that's the direction in which we have to move. And genome and metabolome of those plants are going to help you in doing that, because metabolism is going to give you the products or the compounds or the molecules which will make a food healthy. Now let's understand food. What is food? What we eat is food? No. You might eat a wrong thing. That doesn't become food. Food has to have some characters. Food must satisfy your hunger, but it also must take care of your vital activities of the body or metabolism. Not only that, it should also be something which will help you to perpetuate. That means reproductive system. When something you eat like that, that is food. And if that is food, then what does it do? We did one big mistake when we just concentrated on hunger. That if we make more food, so-called food, then we will be fine. But we forgot nutrition. So we have to understand what is the nutrition. And nutrition you can understand from malnutrition. And malnutrition is not only less nutrition, it can be more than required nutrition also, excessive thing. And if that is so, then you are going to see these type of effects. You will have undernutrition, that is low height for age. You will have underweight, that is low weight for age. You can have vesting, that is low weight for height, although height is there, but very thin people. And then two more things, micronutrient deficiencies, that is real thing that operates your metabolism, and obesity, excessive. So all these come under malnutrition, and therefore your food has to take care of that. But if we see situation in India, this is from NDTV Swasth Banega Bharat data, that 1/3 of the total world malnutrition load we are giving. So that's what we are. We also have to realize where are our weaknesses. And when we start seeing that, I would say that weakness can be made into an opportunity. And this is state-wise, if we look at it, I come from Uttar Pradesh today, and that's the second one after Bihar in terms of malnutrition. And just see the percentage of children who are stunted, first bar, dark one. Then children who are wasted. That means although they are growing tall, but they are so thin or so weak that they won't be able to have a normal life. And then children who are underweight because they are not getting enough nutrition. So that's where we are. So I see this as an opportunity to do research in nutrition and make products. And that we have to follow. Why not? And that why not is a functional food. A food that not only provides the nutrition and the hunger or appetite satisfaction, but also makes your body to not have diseases. Preventive health care. Or sometimes even curative. So can we have food as food only? My chapati as chapati, my idli as my idli, my dosa as my dosa, my rice as rice. But then this is so enriched with molecules that will help me in not having a disease like all metabolic disorders. You know, sometimes we debate, are we developing nation or developed nation? In one sense, we are developed, fully developed. That is lifestyle disorders. We have crossed America. Why? We are not eating proper and we are working, sitting in a sedentary way, not doing exercise, not doing workouts, adding calories, not spending them. That's why we have diabetes, we have nervine system problems, we have obesity, and obesity is the mother of at least 20 diseases. Obesity, I don't mean a fat-looking man, but a man inside fat. The chemistry of the blood, the cholesterol value, LDL, VLDL, HDL ratios, and then triglycerides, which are directly linked with stress. How do we take care of that? So that is where functional foods can really help you. And functional food is functional food because it's rich in the molecules that do that job. And those molecules are called nutraceuticals. So each plant or each crop or each product that you eat has molecules which have the capabilities to do harm or advantage. And when we start looking at it, we can really be looking at nutraceuticals. But you don't have to isolate nutraceuticals to feed the society. You have to have food which must be rich in those nutraceuticals. That means functional foods, and they should take care of what you want to do. And that is your mechanism of action also you would be able to do, and even dosage and toxicity effects you can think about. If we start looking at it from that angle, India is sitting on something which should be a dream of 100 billion to 200 billion US dollars. And this is a report that came up from Nutrify in 2022. And if we want to see, how or why are we saying so? There are two things that India is rich. One is resources and other is ecosystem for nutraceuticals or functional foods. In resources, your university itself is centered on health, farming, and medicinal plants. Now, when we say medicinal plants, when we grow them, we can say it is you are cultivating health in fields. But then you cannot say any plant which is not medicinal. So how can we be choosy about it? But on the other hand, there are eco niches, agroclimatically diverse. I'm not saying agroclimatic zones. I'm saying agroclimatically diverse niches, 52 in India. So literally you can grow everything what temperate countries can do and what tropical countries can do. Active nutraceutical and pharmaceutical ingredients. Now these come from all those crops or plants lower or higher, but 1700 well characterized documented plants are there with India, which contain the molecules for which idea about mechanism of action and how this is going to help in the health is established in our databases, right from TKDL to CBDL and so on. Then ancient grains and ethnic foods which we had left, like pseudo cereals. Or now today we are talking about millets. Even our Prime Minister keeps speaking about it. Now those are forgotten wealth which we had, but we just ignored them after Green Revolution. I never criticized Green Revolution. Green Revolution was the rightmost thing at that time. But we should have also taken care of holistically, like we say in Ayurveda, in agriculture also, that take care of nutrition, take care of diversity on your platter. So that's what where we did wrong. And in terms of ecosystem, we have formulation and processing GMP facilities, enormous facilities. Make in India is happening because of this. And that is for pharmaceutical industry. And nutraceutical industry is not different. It's again on molecules produced, secondary metabolites produced by plants are molecules only. And therefore, same facility which we already have can be utilized for nutraceutical entrepreneurship or industry. And then startup ecosystem, you all know, is doing well in India. And we already have more than 7,000 startups which I would say have translated. In fact, as individual, while institutions are doing great, but as individual, I could raise from 2009 till today around 6 startups who are doing good. So if each one of us starts doing that, just imagine where we can take the society to. Don't wait for any agencies to come. I created Flora Fauna Science Foundation and decided one thing: I'll not take government funding. I'll do it from my pocket and my colleagues will contribute, and that's how we are running. 10 years we have reached, and innovation summits we do, we give awards also. But we feel such a big satisfaction out of that. Of course, your stage is not that. You will come to that stage later. My stage came to give back, and I started doing that. So this can be an era of botanicals. And in botanicals, when we are saying it, what I am saying is adding value to the food, that is food supplementation. You know, we look at supplements in a very different way, like gym goers look. No, supplementation is not an exception, it is a norm when we are talking of nutrition. And now people are not talking about bodybuilding only for supplements, they are talking about general nutrition also. And food supplements, therefore, are a norm, new norm. But those supplements, how natural we can keep them, how we can really be harvesting from flora and fauna is the way to do it. So we should not do casteism in plants and animals. We should not do casteism in higher plants and lower plants. We should not do it between prokaryotes and eukaryotes. We have to look at metabolism, and when we start looking at metabolism, you will find what was the aim of G20? One Earth, one family, one DNA. I say one DNA because DNA is a molecule which is same across every organism that you talk about of flora fauna, with quality difference because of the things coded by this in sequence. And that's why genetic engineering or biotechnology or transferring genes from any system to any system is feasible. I'll come to that in utilizing in our foods also. So I'll take up some of the challenges as opportunities. So one challenge is how can we grow beyond food in farms? So I'm saying cultivating health in farms. So we'll change the spelling of farming. FARMING not acceptable. PHARMING acceptable. So that farming, when we start doing, doubling the income is nothing. You can do 10 times the income. Example, obesity, as I mentioned. Now, obesity leads to high blood pressure, diabetes, blood fat going just haywire, coronary artery diseases, stroke, osteoarthritis, sleep apnea, cancer, even reproductive issues, sexual problems, and so on. So that means if you hit obesity, you are going to be on $1 billion industry. Can you make products from crops against obesity? I'm just giving you, it's a big list, but I'm giving you some examples in each case. Apple has red apple, an apple a day keeps the doctor away. It's because of the pyruvate. Garcinia cocum, Garcinia cambodia, and then indica. We have hydroxycitric acid, which really balances your HDL and VLDL and LDL. In fact, I was in World Congress of Medicinal and Aromatic Plants in Bangkok, in Chiang Mai, in Thailand, and there was a talk before tea, in which a company had come. Company was Hysol. They were producing HCA, and they were showing only pictures before, after, before, after. So all those fatty guys had become slim in their pictures. And after the tea, when the next session started, there was no one here. All were talking to that guy only. How can I be slim? So that's what one single plant can change. And if we go from Goa to Dharwad, it's a rich belt in the diversity of kokum. Lata Mangeshkar's village, Panda, is also on that way only. Then allium, lasun, garlic. See, I was astonished to find in Frankfurt airport smell of garlic all through. So I was thinking it must be a meat product like salami or sausages from where it is coming, but there was no products like that. Then I observed that people were eating the cloves or managing diabetes. So these are the examples, Karela or bitter gourd. It has both the things. The compounds which can mimic insulin. So they are insulin-like proteins, and you have molecules which help you in controlling or regulating your insulin production. You have ivy gourd, kungru, what we call as. You have stevia, which can help you in preventing taking sugar intake, or trigonella methi, where you have physical as well as chemical barriers for sugar and fat metabolism. So you can have a party, have trigonella soaked overnight, have some fiber out of it, a spoonful, so dawat bhi karo or ash bhi karo. You don't have to worry about, this is going to add to your fat. That's what you can do with things like methi. Then nervine tonics. We all suffer from this, isn't it? Not me, all of you. Because you think too much and don't relax. Relax, smile, do the things. And when you do that, you can also add value to your food by having things like bacopa, brahmi, centella, mandukparni, or acorus, watch, or even kamal. I'm not talking of Operation Lotus. I'm talking of kamal. Okay, it has alkaloids which are very useful. So just wanted to give you a glimpse of what medicinal plants can be really doing as functional food ingredients. So future of foods is going to be diversity. You will have higher plants, you will have lower plants, you will have colors and many things. This is your usual food of pulses and grains, and fungi and algae and many other things. So you don't create barriers, and if you start doing that, then you will be able to do that. Now I take another opportunity. This is a conflict, man-animal conflict. Comes from where? Comes from oil palm cultivation. When you replace tropical forests with oil palm plantations, you squeeze the animal population there to a smaller space, and therefore you have extinction of them. And also water requirement is much, much more. And therefore, can we find a solution now? There is a solution which is circular economy itself by shifting from higher plants to lower plants. And the reason is that we cannot get rid of palm oil because huge population in Asia and Africa are dependent for their livelihood on that. 70% of our savory, bakery and other things are dependent on oil palm. And therefore, why to remove it? Why not to produce alternatively? And that is through algae or phytoplankton, where photosynthesis will happen. Like higher plants, you don't have to deforest your forest areas. You don't have to put artificial plantations. You can have in wastelands where you have water bodies and do this production of algae and then producing lipids and proteins both, and you just see the products of the kind they are coming out. Your cheese, your paneer, your So whey isolates are not necessary for a gym goer. You can be dependent on algae. And not algae which tastes so bad. You can convert that into such products. And that's where research goes in. That's where innovation goes in. Why microalgae only? Macroalgae also are very wonderful diet. And they give you fiber, they give you protein, and they also give you biomaterials up to making construction and clothing things. So that's the type of innovations are happening, and private companies are already jumped into it. I'm not reading the names of the companies or those things, but you can read that. And anyone who would be interested in any of the things later can ask me. I can send you the PDF of this presentation. Okay, so don't worry about that. Then food source barriers. We say, ye to yahi banta. This plant produces this plant produces this, or this crop produces this crop produces this. And usually we never look at the plants which are not directly visible to us growing in agricultural farms. But there are ways to look at it. This is duckweed or water lentils, which floats. It's a plant only, but it floats on the surface, and it's a complete food in itself, including vitamins. Even vitamins like B12, which otherwise we say, you don't have, other than animal sources, the possibilities. Here come the possibilities. Have to close? Oh, so I go to last slide? Yeah. How many minutes is it? I'm midway. 31 out of 66. But okay, whenever you say 5 minutes, then I'll come to the last slide. Because I thought I must pass on the information to them, but I'll do that. Fungi can give you meat alternatives. The mycelium gives you the texture as well as the flavor of the meat. And you can make hybrids, or you can replace animal sources from the meat. And then come the smart proteins. Unilever is in a big way into it, where we are shifting from only thought about plant source, that is soybean, into many other things like lentils, fava, wheat, oats, pea, and so on. And those are being converted into kebabs. So that type of possibilities are emerging. There are eggs possible from mung bean. This is a typical omelette made from mung. Okay, and this is a company which has got European Food Standards Authority clearance, and this is called Eat Just company. So they made just egg that looks like egg, works like egg, and if you are talking in terms of the total protein value, we say if many of you would be gym goers, you must be saying that 25%, 20 to 25% is very good protein source in a powder that you just drink and you don't like it, but still, since you have to make body, you are doing that. Here it is 85%. Can you think of the barriers being broken? Why nots and how nots? Healthy snacking should not be, we should not say this is bad. We should make it good. That's the way I feel. And then you have insects also. This is a waste from silk industry, the pupae, which have been made into products like this, the eric food. And all flavors and all those things have gone, and omega-3 is also there. So even lipid-wise, this is a very good product. eggy potatoes, the egg white or albumin that we talk about, expressed in potatoes, a company called Polopo in US is in field trials. And those field trials now are really telling that the egg albumin produced by potato can be used in confectionery to wherever you were using egg white in the same fashion as you will do it from egg as a source. And biofactories in plants. Lettuce is a very good system where you can produce in large amount. Now this company, Pigmentum, was started for making pigments, but now is producing milk in lettuce. Except for some human milk characters, rest of the characters of the milk are there in lettuce, and you can do secondary metabolites there, you can make sweeteners there, you can do vanillin there, you can make pigments there, and so on. Next is opportunity that challenge is limited land and natural resources, and let's do agriculture, no plants, no animals, no land. Can we do that? This is the future of agriculture, cell agriculture. I'm not talking of tissue culture, okay? I'm talking of cell agriculture. That is where you can use the well-expressing cell systems to put the metabolism in them and do it in a very contained condition, something like 100 square meters working like 10 acres or 10 hectares. That type of situation is possible. You could be engineering them and producing through the controlled fermentation the products that you really want. Not only that, now I have no land, no water, no plants, no animals. I make from carbon dioxide and electricity the protein, Solein. Now, Solar Foods is a company which again has got the clearance for making this protein for human consumption. With all those values, that protein will be there. And not only that, you can manipulate here all the amino acid composition there. So all your 8 essential amino acids can be there. And you can again have 80% of the proteins, but you can also add according to the cell system that you have used, not using carbohydrates, but CO2. You are now translating into proteins and products. And those Solein products are now again going into kebabs and things like that. And finally, the precision fermentation, where you can precisely control the fermentation, make what you want in a functional food that you would like to do. Now, I think he's getting impatient, so I'll not talk sweet things, although next was sweeteners. Monk fruit was talked about in the morning, but since he's impatient, I give bitters, right? But bitters also are economically important. These bitters are digestives as well as cocktail partners. And those bitters are big industry coming up. And then, see, we have to save our food, shelf life has to be increased, and there are metabolites in plants which can be used there. Don't throw watermelon seeds. It's a very good thickener for soup. Plus, it gives antioxidants and vitamins and minerals. So your soup, you can remove cornstarch and you use these. You will really love to see that. Banana peel should not be thrown. It's a bacon in itself, plant bacon. You don't have to use pork. You have to use this. You can fry it and peel off bacon-like thing in a sandwich can be there. Ancient wisdom I'll leave because you all are wise. Prebiotics and probiotics, just give you introduction. Probiotics are what are there in our gut, 167 species, 1.5 to 2 kilograms of that. I am moving, 100 trillion cells moving, my own are 10 trillion. 90 is probiotics or my gut flora, and they maintain my health. Your genomics and other things are not going to give answer for health because you are talking about 10% genome, 90% is borrowed genome. And millets and pseudo cereals I already talked about. This is the opportunity. Final thing I'm telling. That this is an incubator I have made in Lucknow. What Dr. Bansal was mentioning, skies, Suman Khanuja Innovations for Enterprises. Here I invite any of the students or young scientists to practice their innovation in my premises without putting any investment into infrastructure, but your consumables you have to afford, I'll allow you free of cost along with the mentorship that you might require to do that. Flora Science Foundation, I told you, is working in villages. We are making e-villages. E is entrepreneurial villages. And I must thank my institute from where I started thinking in this direction, CMAP. There were 600 people. They all were together with me always, and I could make it. The startups that I could raise through my efforts, and now I am into food yoga. Thank you. So chair and co-chair, I'm sorry for taking more time, but you both were VIPs. I was a Aam Aadmi.
Thank you very much, Professor Khanuja, for an excellent presentation. And what we'll have for general discussion after I also make my presentation so that we can open for discussion at that particular time. Thank you, Professor Khanuja, for an excellent presentation.
Sure. Thank you.
So we have Dr. Bansal as our next speaker.
Okay.
And,
So
he has been introduced with university by and
कॉपी नहीं करना
he was
इसी से
talking
करना
about
कॉपी नहीं करना
no sir, copy नहीं किया.
इसी से ऑन कर दो बस खोल दो.
And today he will be talking about display-based sustainable feeding. I request all the delegates sit through this.
अरे लास्ट वाला होगा आज की डेट का.
Only after this session.
अरे आज की डेट का देखो ऐसे कहाँ भाग रहे हो भैया.
And be with us.
आज की डेट का देखो जामिया. अरे डेट मॉडिफाइड में जाओ ना, क्या प्रॉब्लम है भैया. जामिया आ गया ना पहले तो खोल लो बस. आप एक्सपर्ट हो मैंने सोचा इसलिए मैं चुप था. आप ढूंढ रहे हो.
That has been summarized in 30 minutes.
ये क्या हो रहा है.
I would say invite
फर्स्ट स्लाइड खोलो पहले.
experts. Thank you.
शेयरिंग क्या कर रहे हो. ऑनलाइन भी है.
हाँ सर ऑनलाइन.
पॉइंटर ये है क्या. तो मैं यहीं से करूँगा. ठीक है हो गया.
एक मिनट सर मैं लैपटॉप
क्या हुआ.
ऑनलाइन शेयर करना सर.
अरे छोड़ो उसको भैया टाइम नहीं है. पहला स्लाइड ओपन करो एंड आई स्टार्ट. हेलो लीव है ऑल. और आगे कर दो. अरे क्या यार आप क्या करना चाहते हैं भैया. आप बताओ पहले मुझे. हैं. Okay, friends, good afternoon once again, and thanks to Professor Dr. Abdeen for inviting me here once more and have this opportunity to meet you and talk to you all. And this opportunity this time is special because I am able to meet, I mentioned before, my good friend, Professor Khanuja, and also I discovered my another friend, Professor Tuteja, whom he had forgotten me, but I could remember him and we could meet again. Thank you very much. Also, he's an adjunct professor here. And thanks to Dr. Monica for all the logistics and the way she's guiding us all through for managing this session. Well, you have already gone through a wonderful lecture, but I would still request you, all of you, to kindly bear with me and stay back because you would like to, I'm sure, take part as well in discussion later. We heard so profoundly, actually speaking, from a person who has spent his lifetime now, we can say, as a researcher, as a director of an institute, with all his voluntary retirement and now talking about his own way of life and helping us all with functional food, the quality which is important for your good health, and also talking about nutraceuticals and all, which is really very important. There's no doubt about it. We had forgotten, as you very rightly mentioned, during those early days, but that time also he appreciated that, yes, the food was availability of itself was very important. People were dying out of hunger, as we were told, not because they couldn't buy the food. There was no food available, even if they wanted to buy it. So therefore, that time urgency was to produce more food. And in my presentation, I'm going to talk about now that more food has to be produced not by putting more fertilizers anymore, not by giving more and more water as irrigation, not by spraying pesticide indiscriminately, not like that. We have to produce, but we have to again produce more because our population is increasing. We are now the largest populated country in the world. So therefore, we have a challenge that while our population is increasing, the demand is increasing, the purchasing power is increased, there's no doubt, compared to last about 50, 60, 70 years ago, if you talk about it. And there are more challenges, and we have to produce more food than would have thought otherwise 50, 60 years ago. So therefore, friends, what I thought was to bring to you a message right at the beginning, that do we have those technologies now available before us or not? To really speaking, at one hand, meet all those challenges. On the other hand, still, we increase the food production. We have been able to increase the food production, I must tell you as well. But now what we also want to do is we want to accelerate this incremental gain in food production. So therefore, I thought I will talk to you today about one of the latest technologies the world is talking about it, not in agriculture, even in health sector, pharma, diagnostics, and that is CRISPR-based genome editing. And the title which was given to me is CRISPR-based sustainable solutions. Sustainability is the key factor today. We want to increase production. Sustainability for enhancing agriculture productivity. And I'm sure you all aware what are the national goals. Of course, I don't want to go into the details of it. As you know, let's remember the national goal still remains to increase food production as much as we can. And how much we want to increase the food production, some of the figures are given here. Currently, we are producing about 330 million tons. You know, we have traversed a long way. You know, right from, 1950s, we are only producing 50 million tons. So today we are producing 330 million tons, but our requirement, if we want to really happily meet by 2050 or by 2047, as we're talking about it, 100 years after independence, will be about 500 million tons. So that requires a big jump again on a yearly basis. Not only food grain production, even fruits, vegetables, nutri-core cereals, millets particularly, or even milk availability all need to be increased, to meet our daily requirements. And I mentioned before there are certain challenges, and those challenges include like population increase, I mentioned about it, even dwindling resources like land and water, natural resources are shrinking. And again, global warming is reminding us all the time. 2024, you witnessed some of the hottest days. We never heard of, maximum temperature like 50 degrees. Not only in India, in other parts of the world, more than 50 degrees Celsius in Churu, Rajasthan, and in other parts of India. So therefore, global warming is a big issue. And then we have not been, I mentioned before, able to increase, the productivity at the rate we want. 2%, 3% is not enough. We need to really increase that productivity rate. And then we have to all do it sustainably to meet the sustainable developmental goals. If you all remember, the students really speaking must be aware of all these sustainable developmental goals. As Professor Khanuja was reminding you all to do whatever the best you can yourself, what you can do and contribute yourself, not only as a researcher, as a scientist, as a human being, as a citizen of this planet, you can always contribute to sustainable development goals as well. There are 17 of them, and you can find your own way how you can do it, including increasing food production or zero hunger or meeting the climate change issues. And I said before, of course, it's a reminder to all of you that we need to increase production substantially. And then now, just to remind you again, what are those climate risks? This graph shows you some of the extreme weather events in the last about 60 years, from 1960 to 2021 or 20s, and you can see the graph itself shows you the number of such events are increasing. These extreme events, for example, high temperature, or floods, or situation of droughts, and all is increasing. You can see this is a global map, not only for India, I'm talking about it. I'll give you some figures for India as well. And then you can see here, that in the last about 60 years, from 1961 to 2021, already about 21% slowdown or reduced agriculture productivity has already been witnessed. It's not any prediction anymore. In the last 60 years, it has reduced the agricultural productivity by 21%. And it can further get reduced by another 10 to 30%, particularly in rain-fed regions, if we are not able to contain or limit increase of temperature by 1.5 degrees Celsius, or at the most 2 degrees Celsius. We have already, actually speaking, as I was told by some of the experts, already crossed or reached 1.5 degrees increase in temperature as compared to the pre-industrial areas. That was a Paris Agreement by all the United Nations member countries, including India. And now at least they're talking about it. Can we still contain no further increase, and below, can we keep it below 2 degrees Celsius increase? And if it happens, suppose, you can expect another 10 to 30% reduction. So the challenges are even bigger. Not only you want to increase it substantially, as I mentioned before, even you have to meet these curtailing down, which is expected. So therefore, you have to increase your productivity even further. This is the situation with regard to India. On the left graph, you can see the heat waves. The heat waves where the change in temperature has occurred from somewhere between 1 and 4 degrees Celsius. You can see in some part of the country, there is an increase up to even 4 degrees witnessed. We're talking about containing within 2 degrees. And on the right, you can see the impact of this climate change on crop yield instability. Maximum you can see here in the area which are having high impact. These are the high impact zones which are showing. The green impact, green is of course where there's no correlation yet. But other areas in maximum part of the country, you can see there is an high impact of these high in temperature or drought or extreme weather events. And in general, if you talk about it, you can see over the last about how many decades, at least about 3 decades, decadal increase, which has already occurred, is about 0.21 degrees Celsius increase in temperature. This you're talking about the minimum or night temperature has increased already. And therefore it has, helped in a way to expand those arid regions. Expansion has occurred in arid regions, which is not really a very healthy situation. There's an increase of rainfall variability. I mentioned number of these weather events, extreme weather events have increased, and there's increased flooding, which is data from India. Actually speaking, some of the recent data, I'm showing it with you. So therefore, friends, globally, actually speaking, the concept is emerging of climate smart agriculture. Can we do climate smart agriculture? And there are 3 different pillars of this climate smart agriculture has been given here: increased productivity, enhanced resilience, and can we also have reduced emissions, reduced greenhouse gas emissions? Actually, the targeted 0 gas house emissions. So can we meet these 3 different, objectives through the concept of, climate smart agriculture? And what actually speaking, we are talking about it, the climate smart agriculture, the way it is being addressed by different scientists in different disciplines, this is what I have tried to cover it here all. We are talking about conservation agriculture. These are the farming practices, that how these farming practices can help us still produce more in a climate smart way and not having large amount of greenhouse gas emissions and curtailing the temperature under control. And so conservation agriculture, precision agriculture, regenerative agriculture, climate resilient agriculture, and all we are talking about it. These all agronomic terms, there's no doubt about it. They have their own contributions made in the past and will continue doing so. But friends, too important for me looks like that we need to focus more on developing new seeds, and through the intervention of biotechnological tools and strategies. And this particular graph shows you, actually, I borrowed it from Dr. Himanshu Pathak. I'm sure you know Dr. Himanshu Pathak is an expert scientist we have in our country on climate change. He's currently the Director General of Indian Council of Agricultural Research, and also the selected already, the Director General of ICRISAT. So his work, actually, I borrowed this slide from him, shows here very clearly. You can see here this in graph. Though there is a contribution of all these farming practices, related to the climate smart agriculture, you can see a little bit jump or increase in productivity and efficiency of agriculture, but not beyond a certain point. If you want to go, you have to develop new seeds. And new seeds could be developed through conventional means. We continue doing, Dr. Tanuja mentioned about green evolution. That was all, green revolution was all through conventional bed seed products of wheat and rice. Of course, it is even contributing when I'm talking about 330 million tons. It's all again conventional bed crops. But nevertheless, now to meet those challenges, to increase production in a sustainable way, we must try to use, as the rest of the world is talking about it, and fortunately we have these technologies, biotechnologies available. So that particular biotechnology component we need to put in here. And again, as you all know, in August 2024, our Honorable Prime Minister of India announced a Bio-E3 policy, which is, to do with biotechnology for economy, for environment, and for employment. But important for us to actually being recognized here, out of 6 thematic sectors, one of them happens to be on climate resilient agriculture. As you can see here, sector or thematic sector number 4. So therefore, biotechnology is going to play a very big role. And then fortunately, again, we have this technology, though there are others already available, people are using it as listed here. We've got the conventional breeding I mentioned to you before, mutation breeding, which is a matter of the past now, not anymore. Really speaking, people will like to work on it. Transgenic breeding, people are talking about it, 200 million hectares already under GM crops globally. Actually, it's 206 million hectares. In India, about 11.5 million hectares. USA maximum 75 million hectares, etc. But now we have breeding by genome editing. This is another technology. And I'm sure you all know that these 2 women scientists won the Nobel Prize for inventing this particular technology, which we call it as genome editing. And it has got wonders. It's already doing wonders, I must tell you. If you are following this technology, some of the youngsters, some of the students, irrespective which discipline they come from, what we're talking about, of course, we want to have always healthy food. We want to remain healthy. But at the same time, important is also some of your diseases which you inherit. There's no cure, practically speaking. I mean, they suffer, unfortunately. For example, sickle cell disease is one such, cure for which I was told, I don't also keep up as much, but then in last 50 years, there was no cure. But only first time, a product has been developed based on this CRISPR genome editing technology that there is a hope. But of course, it's expensive. But one company has already, which is approved by FDA of United Nations, that is come into the market. And that is through this particular technology. So this technology, not only in agriculture, even in health sector and other sectors, is really making waves. And it takes, when we talk of crop improvement, compared to other technologies, though they are all important, I must tell you, it's not single technology can do wonders. But this technology saves you energy, saves a lot of time. That's very important. While other technology will take about 10 to 12 years. Sometimes if this resource you want to transfer or any gene transfer from a wild species to make it cultivated, it can even take 20 years. The examples where it has taken 40 years. But here using this technology, you can do all that in 2 to 4 to 5 years. Of course, you have to have all that information, knowledge of the target genes, etc. I'll come to you that. And again, fortunately for us, one of the inventors of the 2, Dr. Jennifer Doudna, is talking about it, that this particular technology can help farmers adapt themselves to climate change and sequester more carbon. So she's, while working on a medical sector, finding some of those cures for medical diseases, health diseases, but then she's also talking about, climate resilient agriculture. And therefore, through climate agriculture, we try to, through this particular genome editing technology, we try to develop crops with enhanced heat and drought tolerance and climate resilience. We try to develop crops to fix more carbon dioxide so as to increase carbon sequestration in the soil. We reduce greenhouse gas emissions from agriculture, and also we develop crops which will still give you more production with minimal use of these water, nutrients, fertilizers, and pesticides I mentioned before. So while conventional breeding, of course, is focusing on increased productivity, which is very important to some extent, resistance, etc., but we are not able to have a major breakthrough with regard to minimal use of these natural resources, or increasing the use efficiency of these natural resources like water, like nitrogen, or even without use of agrochemicals. We're trying to talk about it here, and for ensuring finally sustainable farming. So I give you some of the examples using genome editing. Time will not permit me to go into the details of it, but only a few examples. And before I come to the talk about science, which we are doing in India, in various labs. So this is one of the examples in rice, early examples when CRISPR technology was used to silent some of the ABA receptor genes. There are about 12, 13, 14 of them in rice. They did silencing of using CRISPR technology in one by one, or kind of permutation combinations. They found, particularly as shown here, of this ABA receptor 1, 2, 3, 4, 5, 6, they all did together, or only 1, 4, and 6 gave a better growth and productivity, which was not known before. while they are able to provide stress tolerance, this particular hormone, but it is also associated, it provides a balance between stress tolerance and growth and increase in productivity. As you can see here in the graph, the one which is wild type versus the genome edited, yield under natural conditions in field in China. We know the scientist, Dr. Jiakang Zhu, is our good friend and collaborator. He's been working and wonderful, good work he has carried out. So in China, under field trials, they have been tested, and it is giving you an advantage about 30% increased yield, and also a good advantage under stress conditions. Another example here is right from the institution. We call it as Innovative Genomics Institute, which is new, which was established by Dr. Jennifer Doudna with the money she got of the Nobel Prize at the University of California in Berkeley. That institute is also, I said before, contributing to climate resilient agriculture. They have come up with this kind of a strategy and technology wherein they are able to reduce the number of stomata. Some of these plant physiologists, scientists, they will know that water is lost when they the plants take up through root system, which is essential. Water is taken up and it must be lost to the atmosphere. But where can we control it? And that all happens through stomata. So idea is, and because stomata only allows the CO2 to come in, so we have to strike a balance that it will lose enough water which is required to maintain the temperature of the leaves optimum, but also not inhibit photosynthesis by curtailing the CO2 uptake. So they have come up with a strategy where they can optimally reduce the stomatal density, so that the plant growth is not affected, and instead they are able to conserve water. So this is one of the genes. They actually tested 2 genes. With one gene, the stomatal density came down drastically. That was no good use, because you want to have it optimally reduced, and that's what they've done it with one of the genes I'm mentioning here. It is, epidermal patterning, gene. We call it as EPFL10, and which is a negative regulator of stomatal development, positive regulator of stomatal development. And they cut down the expression of this gene using SDN1 technology of genome editing. And you can see these lines, the ones, either of the leaf, here the 5th leaf, they've got less number of stomata compared to the wild type. Here again in flag leaf, they've got less number of stomata compared to, but that was enough for the plants to conserve a lot of water and moisture. But quickly now turning down to another about 10 minutes, I thought if I'm able to make a story complete, these all genome edited plants, people thought should be regulated as GMOs when it started, let's say about 10 years ago. But many of the countries that you see here in graph shown in green color, they decided genome edited crops without foreign DNA, should not be regulated as GMOs. On the right hand, you see in our country alone, this is an example from India, all GMOs, particularly GM crops, they are regulated. There is a pathway. We go from one IBSC, which is in your own institute, to RCGM, to GSC, and it takes 8 to 10 to even 15 years, which is a very unfortunate situation, I must tell you, in our country. It has taken even 20 years at times, which is not a very healthy situation when we talk about it. Because based on science, all tests have been done, for example, in a crop, the tests which were all listed. But anyway, apart from that, the point of which I'm trying to make is that should we regulate genome editing, in plants as well or not? You can see here this, I'm showing the, regulatory status in the world in 2020. About 10 to 12 countries shown here, they decided not to go for, these biosafety regulations if there's no foreign DNA present, right? India, the map is yellow. Why yellow? Because we have not decided either way yet. We are in discussion. And when we got a document, Government of India prepared, Department of Biotechnology, we came into picture. We came into picture as a National Academy of Agricultural Sciences, of which Dr. Khanuja and many others happen to be important, esteemed fellows. So we had a thorough discussion actually in our academy to decide on that particular document, whether we should be regulating, such a, technology like GMOs in India or not. And finally, there was a landmark decision. I would say very clearly because of the efforts we scientists made. We were very, very clear about it, the science part of it. And we in 2022, we succeeded on March 30th, 2022, we got this landmark decision that government allowed exemption of these genome edited crops, particularly of 2 categories. I can talk to you students about it details later, SDN1 and SDN2 from biosafety assessment. And we fortunately developed standard operating procedures. I happen to be a part of it, and these everybody would like to see. Those who are really interested in working on this technology must go through it. They're very simple. When we were meeting, number of times, we wanted to make it simple and subtle. And that's what it is, actually speaking. World is appreciating it. I've shared this with many other countries on demand. And after we succeeded in March 2022, soon after this, then we decided, group of us, that what should be now India focusing on, what crops, what traits, through particularly these genome editing technologies. So we came up with this kind of a strategy, and we published it in July, just 3 months later in 2022, in an Indian journal, Current Science, wanted to give a roadmap. And in this roadmap, then we developed a project, a network project on 5 crops, thanks to ICR and SF, to give this project, not to me. I happen to be in the committee, so therefore I'm very excited about it. Dr. Vishwanathan is the PI, and 2 such products already genome edited are undergoing field trials. They are exempt from biosafety assessment. They have followed all those SOPs we developed. They're already undergoing, for the last 2 years, field trials. 2023, 2024, it will now enter 2025. And Honorable Minister at that time, because we convinced them as well that there should not be any biosafety regulation attached to these technologies at all, he was excited to give a statement in the parliament. By 2026, India will have the first genome-edited rice varieties. And these are the 2. One is, which is saving 25% of water in rice. Another is, which is giving you high productivity of 25 to 30% in rice, from 2 different institutions, from IRI and from Rice Research Institute in Hyderabad. Then to move on, we added more crops. India has decided to work on 40 crops with rupees about 500 crores of budget, which is only starting, I would say, few months ago or a year ago, at least, let's say in 2024, made of it. And the efforts are being made by several institutions. Here, again, the traits of importance are high input use efficiency, which is otherwise not easily possible through conventional breeding, or even through transgenic route, or climate resilience, increasing tolerance to all these stresses I mentioned before. But you know what? When we were preparing that paper to identify not only the crops and the traits, we also wanted to identify what are those target genes, because in genome editing, you need a target gene, which you would like to silence or make it correction into that gene through genome editing. So when we were going through that kind of analysis with all the breeders in at least 10 to 12 identified crops, in majority of them, the major bottleneck which came was the availability of these suitable right candidate genes, which could be targeted. So therefore, I have my own experience of working on plant genetic resources. So we thought it's important to have this characterization evaluation. I thought of it even before we talked of CRISPR editing, because anyway, for breeders also, it's required to use those resources in their breeding programs. So we decided to go for characterization evaluation. It's very, very important to identify the target genes, and also do functional genomics. Actually speaking, functional genomics are required in those identified, natural genetic variation is there in that material, to identify those target genes. And you know what, again, I'm very proud to put before you that India is home to the 2nd largest gene bank in the world. Whether you're working on plants, microbes, okay, or in animal sector, or in the health sector, you must know the genetic diversity is very, very important. Dr. Khanuja also mentioned about it as a presentation of the kind of a variability which is there across in our different tracks in India. So here I'm saying you that we are the 2nd largest gene bank in the world, and we have got collected large number of wild species which have been also put together with the kind of excellent traits they have in them. And then, important is, while we are very good in conservation of all these, therefore we are the 2nd largest gene bank to conserve. But not only we have to conserve, we have to characterize them now for identifying the target genes, we have to evaluate them. So we took a challenging task, and that's how, the company matters a lot. I was friend to Dr. Suman Khanuja. So anyway, naturally, our thinking will also be different. You would like to be more innovative. You would like to really do things in a different way and different style. So we wanted to do here first time in India again, or maybe many of the institutions, to characterize all the germplasm available in a crop, for example, wheat, which is very important, which is not easy task. About 25,000 accessions. We removed 3,000 duplicates. 22,000 accessions of wheat were characterized in a one go at different locations for various, traits, in wheat. I don't want to go into the details of it. In fact, it came into Limca Book of Records We got papers which were covered on the cover of Crop Science, which is a very good journal. Academically, we all know about it. And we could identify the kind of genetic variability which we had never seen before. Importantly, with regard to climate resilience, we identified some of the accessions, out of 22,000, about 3,000, based on certain parameters, which were very important material. Now, this is the material which we can utilize for isolating those genes and targeting those genes and mimic that genetic variability through genome editing into high-yielding cultivars of wheat, for example, in this crop. And then, as I said before, we were doing this paper on for current science. And here I'm just giving you one slide in case of pulses, like chickpea, pigeon pea, and green gram. You can hear we identify these prioritized traits, but when we're looking for potential candidate gene, either for SDN1 and SDN2, in majority of the cases you see here, genes are not even known. So while the technology is ripe enough to be utilized, but we are lagging behind in characterizing, evaluating the natural genetic variation, going for functional genomics, and now people are even talking about going through, more pangenomics. So what we thought initially to again characterize the entire collection, about 14,500, in case of chickpea, we developed a core. And again, we were collaborating that time in 2012 to 2013 with Professor Rajiv Varshney. He's a household name, Professor Rajiv Varshney. Should invite him one day if he's available, actually speaking, who is making waves at the international level when you talk of genomics in any crop for that matter. So we collaborated with him, came up with the first paper in Nature Biotechnology on the reference genome of chickpea. And then recently, 3,366 genotypes of chickpea have been sequenced all or resequenced, you call them, And then new genes, this is called a pangenome. And about 1,500 new genes potentially useful for various agronomic traits have been identified. Some of the deleterious genes, which are causing disease, for example, or which are, negative regulators of plant growth and development, they have been identified because those are the ones which can be now corrected, or deleted their function through SDN1 and SDN2 using CRISPR technologies. And so therefore, what I'm trying to tell you here, friends, that while we have these excellent genetic resources, here's a roadmap how you can capture this, genetic variation and install that through genome editing into your already elite varieties. Not that you do it in any variety. No, it will not work. That's not required. And therefore, you further develop a new improved varieties. And when you talk of plant genetic resources, one of the biggest treasure we have in India is on wild species. Any country will have it for that matter. We are fortunate. We have a large repertoire of some of those wild species, for which we have characterized and put them all information together. So while we were trying to increase high yields in modern breeding varieties, there was a reduction in genetic diversity, and that reduction in genetic diversity leads to poor adaptability of these modern cultivars to climate change. So we need to look for those genes, and gene editing is the technology and the way forward. And fortunately, again, I mentioned before, earlier I mentioned if you want to transfer a gene from a wild species to a cultivated type, high-yielding type, it will take you not less than 10 or 20 years. In IRRI, there's an example in case of particular wild species, it has taken even 30 to 40 years. But fortunately now, it's hard to believe the same thing you can attempt using genome editing from wild to cultivated type in 2 to 4, or even if it is 5 or 6 years, I think it's worth it. And all those wild species have those genes for sustainability, climate resilience, doing better with less fertilizers and agrochemicals, etc. And which we lost those genes when we're talking about, domestic or conventional plant breeding, developing modern varieties, etc. So friends, therefore, take-home messages for some of you who would like to take this as a challenge. You have already been charged enough for taking the challenges by Professor Suman Khanuja. But here is another kind of a message for you all, that gene bank functional genomics is required to be done. Develop efficient delivery and genetic transformation system and elite crop varieties, not in any variety which you would like to do. This is easy to do, no, only an elite variety, or develop such a system in wild crop species, and also develop an efficient DNA-free or tissue culture-free or DNA-free, RNP-based. We can talk about it later, and then develop a viable public-private partnership. You can do all alone. You must bring in the private partnership. They're good in certain things, and also, of course, capacity building and human resource development for some of the youngsters. We're organizing regularly. I'm organizing very regularly some online, offline, training programs. I'll invite you all if you would like to be part of it. And fortunately, to realize the importance further, of gene banks, I'm sure you all know about it. In just recent budget, a month ago, Government of India has announced another gene bank for safety duplicates, we call it, to conserve 10 lakh samples, which is a very big thing. Only one we have internationally somewhere near North Pole. I had an opportunity to visit that in Norway, but this will be the second one. We'll have it in India. We want to conserve our own germplasm, actually speaking, which we have in large quantity. So therefore, with this, I thank Dr. Vishnathan, with whom I keep collaborating. I thank Dr. Kutub Mola, one of the scientists who has been actually awarded by Flora and Fauna Foundation as a young scientist, Professor Sushil Memorial Award Lecture. Award he has been awarded. These are my two very close collaborators. And of course, I also thank my whole institute when I was working in NBPGR. These all scientists who collaborated with me to do all that big exercise of entire characterization of wheat and chickpea, which was not easy at all, I must tell you again. So I'm very thankful to all of them. And of course, thanks to ICR, to DBT, to National Academy, where I worked in in different capacities. And I was fortunate, and I must thank Dr. Jennifer Doudna, the Nobel laureate, who invited me. Actually, her institute, I had an opportunity to visit this Innovative Genomics Institute last year, January, in 2024. So I must thank her. I keep interacting with them. Their scientists from IGI recently was in India only last week. 14th of February, they've gone back. They are here for conduct some practicals, for training workshops, etc., on genome editing. Many of the student scientists participated, and so I must thank her as well for the kind of leadership she's showing at the world level on genome editing for climate resilient agriculture. And they're helping us, they're helping India, and they're helping around many other countries of the world. So with this, I thank you all for your patient listening. If there are certain questions, we'll be very happy to take those questions. Thank you again, all of you. Thank you.
I will request you to please join us here.
I will join you there, but I think what we can immediately use effectively 5 to 7 minutes time is because I know we are running late, and the speakers for the next session, they're already here, and we have to also eat our lunch. And therefore, I would only still request you, if there are no questions, no discussion, we want to just end up here. 7 minutes, if we can have effective discussion, 7 minutes. We must keep it. It's a large audience, and so that we break at 2:30. Is it okay with the floor? Right. So therefore, now open for questions. Anyone, any question to any of the speakers, both of us, or even, to the speaker of the next session, whom you have not heard yet. Questions, please, from the students. Otherwise, you will think you are not listening. Or from anyone. To Professor Dr. Khanuja. Yes, please. Quickly, Mike, jaldi se dijiye. Time hame save karna hai. We would like to have at least 5 questions, if not more. Yes.
My name is Rashid, and I'm a second year biotech student. I have 3 questions, one for you, sir, and for you. First,
You have got what? 3 questions?
3 questions, yes.
Ask one. To Dr. Khanuja, ask one to me.
Okay. You said that the availability of suitable tablet genes is a bottleneck that is keeping us behind in this race. What, according to you, and what challenges do you think we are facing, and what makes a gene a suitable target gene for incorporating in?
As I said, we need to characterize, evaluate the natural genetic variation which is already occurring. While we're talking about even using in our modern crop development programs to conventional breeding, some of the modern varieties, their genetic base is very narrow. We must move away from it, because then only we can try to get those genes which are important for enhancing nitrogen use efficiency, or for adapting to various stresses for climate change perspective. So we need to explore those. We need to genomics. We need to do function genomics, and there are tools available plenty. Only thing is that material needs to be utilized, and using those tools, and you can identify those target genes. Then you can test those directly into any variety. Which germplasm you have already done that. Test it, and you can do genome editing. You need to develop, of course, important, not only having our target genes, to need to have, so far, unfortunately, we have no tissue culture free method available, though people are working on it. At least, I must tell you, 5, 6 such strategies being worked out, but we need to develop through tissue culture, so therefore that protocol also needs to be developed.
Thank you, sir.
Sure.
You mentioned that there are many things.
In the meanwhile, I request others to kindly think of question, because I'm curtailing his question down from 4 to 2, only because I would like to give opportunity to others. And yes, please, quickly.
Yes, sir. So you mentioned the different agriculture, cell agriculture techniques that we can utilize to create different sources of proteins, and which one you mentioned, if I'm correct, yeah, 85% protein. Has there been any research in the bioavailability of those proteins? Because these 85% protein, even in some food crops, when they have protein, it is not 100% available for our body to utilize it.
The bioavailability is the first criteria when we talk of food, right? So those people are not saying that you eat 85% of the protein at par with what you are eating as a daily aspirate. What they are saying is your requirement is going to be reduced. So if you are taking, say, 100 grams of that protein powder, they will be 10 grams of that protein powder. Obviously, as I told you that even balance of amino acids that is important as far as the nutritional part is concerned. To identify it is commercial.
Thank you, sir.
Okay, another question, please? Yes, go ahead quickly. Speak loudly, don't wait for the mic.
I would like to ask that if we are increasing these yields at a certain amount of rate, would there be any negative effects showing later in life?
What is in your mind? You have any negative effect?
So, like alteration in the genomes or some other effects, like in.
So if there is an alteration in the genome, it is not done at random. Using this technology, particularly I'm talking about it, it is done in a precise, accurate, predetermined fashion. And even if the changes were made at random, never mind, we have been doing all through, we continue doing so, but we finally select the combination of those genes, through a process of generation after generation, kind of, growing in the field, the ones which are more stable, which are more morphological suitable, which are more high yielding, which have no negative kind of effect on any other parameter of the plant growth and development, and therefore it takes time, 10 to 15 years, and we stabilize that material. There is a whole process of conventional plant breeding based on such things.
Yes, my next question, sir. As we all know, CRISPR-Cas9 has many off-target effects.
So I understand your question. To save time, I will not worry as a scientist of any off targets. Let they be there. In plants particularly, I'm talking about it. And therefore, you kindly go through our SOPs. We have not mentioned of any such information we require from a developer on off targets at all. Why so? Because there is a science behind it in a way. Or we can say we have learned enough from our experiences in the past. How many of our experiences? At least 80 years of experience, 80 years. 1940, we started with chemical mutagenesis, okay, to have genetic variability. We started with irradiation. Nobel Prize have been won by those who were able to show all this, right? We have been doing this random mutagenesis. 3,000 and more varieties are available for users around the world, right? United Nations agencies are working on it. And there's no ill effect. Why? Because we have only selected out of those random mutations, it takes again time, only those which are morphologically superior or kind of optimum with high yields, etc., etc. So there's no problem at all. Here, off targets, they are not. We select finally the phenotype, which we have intended to do genome editing for that particular trait. If there's some abnormalities, we discard those. And we only take those plants which are forward. In majority of the cases, though even if they're off targets, you find the ones which are mostly suitable to your conditions for which you have developed it, only we take those plants forward.
Thank you, sir.
Thank you.
I would like to ask, sir, we are increasing the protein and other concentrations in the nutraceutical. Sir, what is the outcome that our bodies are metabolizing those increased yields?
What you are doing is that instead of eating 100 grams, now you are going to eat 10 grams.
So, but our intake rate would be like for 100 grams only, But
like that is according to your requirement you are going to take. When you are requiring, let us say, 105 grams of protein, right? And from one source it comes from 1 kilogram, and another one it comes from say 100 grams.
Is it okay?
Then in your platter you are able to arrange it. Right? What you have been doing in terms of isolates and other things that you are doing the shaking and eating it, and it's not bioavailable. bioavailability there is not 100%, right? And that is why what happens is that you are taking more protein than what you are requiring to have something which your body would utilize to develop only muscles, right? But here we are talking about a functional food where you are going to have its availability as well as palatability both taken care of. So that's where the difference would be. And when we make that, let us say there is a neatly in proteins, right? Now you don't have to add another thing. You are already having sample which is going to give you some protein, and your Italy or mixture is having some protein, and ultimately your RDA, recommended daily allowance, that is going to you cannot have any nutrition from one single source. Never try that. If you do that, then you could be going like celebrities are going. Their skills are not good, their metabolism is not working. Those who are bodybuilders, you just check all those celebrities.
Right.
They are having suffering from digestibility issues and pain-getting bone syndrome. Right? So we have to take care of that. That small is beautiful and is.
Okay, thank you very much. I think it's 2:30 and we close the session here and we give big hand to both the speakers. Over to Dr. Monica.
Thank you, respected chairpersons and our esteemed plenary speakers for your insightful and thought-provoking contributions to this session. I now request the chairpersons to please felicitate our distinguished speakers. May I first request Professor Suhail Parvez sir and Professor K.C. Bansal sir to please present the memento to our very first speaker, Professor SCLS Thanuja sir, founder and chairman, Flora Fauna Science Foundation. Now, I would request Professor Thanuja sir and Professor Suhail Parvez sir to please present the memento to Professor K.C. Bansal sir. Thank you so much, sir. To express our gratitude to our esteemed chairpersons for their invaluable guidance and contributions, I now invite Professor N.C. Dardin, sir, to kindly felicitate our chairpersons, Professor K.C. Bansal, sir, and Professor Suhail Parvez, sir, with the memento. Thank you so much, sir. Now I request chairpersons to please present the memento to the moderator of this session, Dr. Monica Selvi. Now I request all of you, sir, to gather for a group photograph. Now, this is the meal time, so proceed for lunch in lobby area, and we will be resuming here with our plenary section 2 after 30 minutes sharp. Please enjoy the meal. Thank you.
Okay, ma'am. Thank you. We will in the same way.
Yeah. So I don't say that any of the question is irrelevant. All the questions are relevant, and it is not true that scientific authority is opposing or anybody is opposing for that matter. But the demand of the situation is you need to have a newer scale of everything, a newer scale in terms of generating the associated data, associated knowledge around a product, and a newer scale to have a system to judge those data and take a decision. Our scale is small. That is the one-line message that, as per my opinion, the scale has to be multiplied in a larger way. Otherwise, all the questions are pertinent. It needs to be addressed, and it is only after addressing these questions anything is decided in any other country. That's absolutely fine. But our scale has to be really increased.
Can I ask another question?
Let somebody else ask. Anybody else? So, you?
Hello, sir. First of all, I congratulate you for the very good insights about the transgenics.
Thank you.
It's just that in case of outbreak, if there is an outbreak, transgenics needs a lot of time to be prepared. How scientists will make such kind of advancement to target such kind of outbreaks in that situation?
Yeah. So, there are two aspects. If you recall the examples that I have shown, the outbreak came 20, 25 years before. So they were struggling with it. But the moment a solution was visible, it was adopted immediately. So receptivity towards new technology, that really made a difference. If you see the example of that potato lead blight, as I said, the Irish famine because of outbreak of potato lead blight happened way back in 50s. But the products developed maybe some a decade back. It took time, but the important thing is once you know the problem, you try to manage with best available things, and by the time you find a solution, better solution. And then another important thing is receptivity towards new things to adapt. That makes difference.
Okay, thank you. We have got of time. So, Dr. Bhattacharya,
Thank you.
everyone. And please, Dr. Bhattacharya is going. So, Architects, if they want to be now on. Javed. You want to give next round or is it on?
इसे हटा दीजिए
अच्छा
पहले से बताना मेरे क्या है सर वो प्रेजेंटेशन है कर दो
ज़्यादा जरूर दीजिए
ठीक है नेटवर्किंग क्या है ये बंद कर कोई ये बंद कर देगा कि इसमें प्रेजेंटेशन समझ रहा है इसके बाद सर को बोला जाएगा लगाई थे ना
So the next speaker is our co-chair, Dr. Chakravarti, will introduce himself and say, sir.
सर अपने आप को इंट्रोड्यूस कर रहे हैं क्या फायदा
This is JHU, it was written.
Yes, sir.
Is this written JHU?
जी सर जी जी from that बस ये बोले रहने दीजिए 5 presentation तो कर दूँ ना उसको मैं हाँ वो प्रेजेंट कर दूँ I can. So. So it was meant for slide change. LED was not space and pointer and forward and back really. This is for what? This is actually we have shortage of ports. That's why that's not it. अच्छा हाँ यार वो चलेगा नहीं वैसे it's not pointed because of shortage of ports. We can do from here. This one? Yes, sir. This one. This one also? Yes, sir. Yes, sir. It's down. It's down and okay.
Very good afternoon to all of you. First of all, I would like to thank the organizing committee, Professor Abdin, and faculty staff. They never forget to invite me whenever there is a conference in Jamia Hamdan. I'm really thankful to you. The chair, the faculties, and all the students. So my name, the co-chair told already. This is my name, and the institute name is National Institute of Plant Genome Research. Can you hear me? So I don't need microphone. So this is in JNU campus, and my group actually work in proteomics. How many of you know about proteomics? The study of protein is proteomics. When you study one or two protein, then you call it protein biochemistry or protein chemistry. But when you study hundreds of protein from a cell, from an organ, or from an organism, that constitute the proteome. So my group always work in two different phases. The first one is discovery proteomics. The definition, the term itself will tell you that discover or identify a protein which is of great importance. The second is translational proteomics. You identify 150 protein, 200 protein. So what? So you picked up some hypothetical protein, a protein of unknown function, and then study and tell that I have identified a protein which actually perform this job. So with this, let's go. I have only a request. Except the first slide, all my slides are just prepared and the paper is under revision, so don't take any photo. Exactly 25 years back, in the year 2000, I started my laboratory with this group of students when there was no laboratory in India who actually started proteomic work. So I have started proteomic work, and I have decided that I will be doing organelle specific proteome, compartment specific proteome. So in one liner, fractionate the cell and take out from cell wall, cytoplasm, mitochondria, nucleus, etc. You name anything. So this list of papers will tell you that we have dissected almost all the organelle in a plant cell and identified those proteins and utilized also making it transgenic knockout system to prove what is this function does. One might ask a question, why did you do actually organelle proteomics? Organelle proteomics, the importance is you never understand the protein function unless you know where it localizes and what type of function it does. It's simple as that. So with that, if you see this slide, then you will understand from the outer part of the cell to the interior up to nucleus, we have studied all the organelles. Two organelles are still left out. One is Golgi complex and another is the largest organelle, that is vacuole. Today, I am going to talk to you about mitochondrial proteome. This is the plant mitochondria. In rice plant, isolated, purified, and in the year 2019, actually we dissected what the mitochondrial protein does in rice plant. So this paper was published in 2019. For the last 4-5 years, I have been working with only one mitochondrial protein, and the story today I am going to share it. So this part is discovery proteomics, and the second part I will talk the translational proteomics. Sir, whenever 5 minutes will be there, then please give me a signal. I believe I will be able to finish before that. Okay. Now, this is rice plant, 4-week-old rice plant. This is control. This is 3-day under dehydration, 6-day under dehydration, and 9-day under dehydration. Mild, medium, and severe stress. Correlated with the physiological trait of rice plant. All sort of physiological, starting from photosynthesis to transportation, loss of water, lipid peroxidation, etc., etc. But look at and correlate how much damage is done to the mitochondria. So if you look at this, and this, you will understand that as the stress continues longer time, the damage is more. So we gone for proteome analysis. Now the proteome platform is this, different stages, a control, and then 3-day, 6-day, and 9-day stress condition, and we use iterate based quantitative proteomics in a nutshell. Then what happened? First you isolate intact mitochondria for any organelle. If you want to do organellar proteomics, as much as purification step you follow, the better the result. So when we isolated the intact mitochondria stained with rhodamine 123 stain, you can clearly see these red spots are intact mitochondria. Again, how do I biochemically know that these are actually intact mitochondria? In the intact mitochondria, the COX-2 function, cytochrome oxidase, is a marker of mitochondrial protein. You do not see any function here, no activity. You put a pinch of detergent and study it, it shoots up the activity. That means the mitochondrial membrane was intact in the intact mitochondria, and then you can also study the enrichment by immunoblot analysis. Take COX-2, again the same protein, antibody, you see the presence of mitochondrial protein, and negative control is Rubisco, chloroplast protein. There is no Rubisco. However, cytochrome C oxidase is planted. Conclusion is that we have isolated intact mitochondria, and from there we have isolated mitochondrial protein. Now, the way I show iterate-based proteomic analysis, these are the results. The first Venn diagram, it tells the upregulated protein, and the second one tells the downregulated protein. One important message I want to give you, you look at this place, you see only 6 proteins are common. All other proteins are stage specific, maximum either upregulated at 6-day or 9-day. But common protein is 6. Similarly, the downregulated proteins, only 2 are common protein. All are stage specific. The entire data was interrogated by 4 different programs. The names are here. I tell you conclusively that more than 82% proteins are found to be localized to the mitochondria. Now we are happy, and the result is 101 protein were classified as differentially regulated or differentially expressed, and the entire profile, expression profile, is listed here. Finish your discovery part. So we identified 101 differentially regulated protein under stress condition. As I said, now you go to the translation site. What we can do with this wealth? Identification of protein is just wealth. Now, where to encase that wealth? This was the photograph just previous slide I showed. Look at this. We saw one protein give a deep dive. Deeper dive you give, study several days that what we got differentially regulated protein. We found a protein is highly upregulated across the stress time point, and 6 hours it is the most. But the database showed it is a hypothetical protein. That means its function is not known. Looked at the sequence, peptide sequence. We found that protein houses a domain. It's called domain of unknown function, or short form DUF. So we thought we will characterize this protein. Why it is unknown in the literature. So we named it OH-DAF1, Oryza sativa DAF1 protein. We cloned it. Now, although we have identified this protein in mitochondria, in organellar proteomics, the very important question is whether after doing so much purification and study, still we ask this question, actually it is a mitochondrial protein? Now, these images tell this is really a mitochondrial protein, predominantly expressed in the mitochondria. You look at here, the circle tells that this protein is clearly localized to the mitochondria. When we studied the transcription profile of DAF1, then we found along with dehydration, it also induced under oxidative stress, methyl biologen, hypersalinity, also biotic stress like salicylic acid and jasmonic acid. Conclusion is that we identified a protein which is not only responsive to dehydration, but also responsive to multivariate stress condition. So we found it is a suitable protein, and let's go take to the next phase. So in doing that thing, we immunoprecipitate its interacting partner, because if you want to characterize a protein, telling that this protein does this function, hardly there are any polypeptide or a protein itself make a function. It needs some partner, some complex. So we found by immunoprecipitation, in the immunoprecipitation, 28 proteins we found by mass spec analysis. And one of the protein we picked up, that is peroxyredoxin or PRX. When we looked at the peptide sequence, we were a little surprised. We found that the PRX is a protein which is localized to the chloroplast. Now the confusion. How you tell that a mitochondrial protein function in combination with a protein which is actually expressed in the chloroplast? We were confused. But me and my students are so lucky that when we cloned it, we had given for sequencing 2-3 clones. And then we found that sizes are different. It is the same protein, but the sizes are different. Why? We understood that this is an isoform. All of you know that isoforms are generally the product of same gene, but sometimes spliced away some of the nucleotide. So we got one full length, another is smaller size. So we named it OXPRX 1.1 and OXPRX 1.2. Very significantly. Now you see the localization. Actually, PRX1 localized to the mitochondria. However, 1.2 actually localized to the chloroplast. So for us, 1.2 is not important. For us, 1.1 is important because now we found actual partner of DAF1. Now the conclusion is that we identify a DAF1 protein, an interacting partner is PRX 1.1. So this, up to this slide, this is the summary. And the physical interaction, all of you know, that the two proteins are interacting partner, and the physical interaction can be done by many ways, and one of the best way is yeast-2 hybrid. So this is the result of yeast-2 hybrid, and you can see clearly that PRX 1.1 is a true partner for DAF1. Now, what do you do? Where the good protein, bad protein you get, you have to introduce it either in a heterologous system, or in a native system. The best way is that if you have standard transformation protocol, then you take it to the native system. So DAF1 was overexpressed in the background, rice background, and this rice actually is a hypersensitive cultivar. Now you see the result. So what we did, these rice plants were subjected to dehydration for 13 days and then allowed to grow after 7 days, re-watering the plants and see the recovery stage. This is control or vector control. Now the plant, more than 85% plants actually revived. However, the vector control could not revive. So this is the phenotypic. Another important thing that when you put these plants under stress condition, DAF1 increase and also PRX 1.1 also increase. So this suggests that the induction of DAF1 accelerate the activity of OX-PRX1. So they synergistically actually perform function. Another thing that this was a mitochondrial protein. So under stress condition, mitochondrial dysfunction is there. Now, if the plants are surviving better, probably the mitochondrial dysfunction revert back. How you know that mitochondria now is functioning? We did battery of experiment, but I will mention only 2 experiments. All of you know that mitochondria are the powerhouse. Why it is called powerhouse? Because it produces all ATP. Now, when the DAF1 overexpressed, then you see better ATP synthesis. Also, second point is oxygen consumption is now in the up. So 2 things happen, that mitochondrial dysfunction goes away and you get ATP synthesis, as well as that oxygen consumption is actually increased. At biochemical level, now you can see that DAF1, how DAF1 helped the plant to survive stress condition. Survive by producing more ATP as well as oxygen. Two things are there. When plants undergo dehydration, it overlaps or crossover with oxidative stress. How do you know? This simple staining actually tells. NBT and DAP staining. I'm sure that many of you guys do this type of work, NBT and DAP staining, and you can see that where DAP1 is induced, actually the accumulation of ROS goes down. Why? Why that all the oxyradicals goes down? So we studied the transcriptional regulation of all the ROS related genes. You see, ascorbate peroxidase, catalase, SOD, and you can see in the overexpressed plant or transgenic, all these enzymes are very high. That tells how they take care of ROS production, or in other word you can tell that actually it induces the catabolism of ROS much faster than in the vector control plants. So gene or protein can be characterized by 2 different ways. One is class way, that means you put and make transgenic, and another, since morning we were discussing here, I listened one lecture. What is the second one? Second one is knock out the gene. That is called loss of function we approved. So what did we do? In the same variety, we knocked it out by CRISPR-Cas. What happened now? In the transgenic, when the plants become resistant to stress condition, when you knock it out, that one, the plants become hypersensitive. So this correlate this one thing that you overexpress it, it becomes resistant or resilient. The moment you knock it out, actually it becomes hypersensitive. So we, together with all these results, I can claim that we identified a mitochondrial protein in rice with a domain of unknown function and proved it that it helps or promotes the resistant character to plant, and proved it either by transgenic or by, now finally, can I have how many minutes?
Yes,
10 minutes. Okay. So I said that I will be able to finish by the time. Now, since I told you that PRX 2 forms are there, still confusion is there, 1.2. PRX 1.2. Now to negate that in no way the function I am claiming that DAF1 giving plants, it is not the chloroplastic peroxyredoxin. This is one thing. Second is that no way DAF1 has any other chloroplastic partner to perform this function. How you can do it? Only way you can do it, you pick up an organism where chloroplast is not there. So what we did, we have taken Saccharomyces cerevisiae, where there is no chloroplast, and let's see what happened there. This is what happened. Independently or in combination, you introduce DAF1 and PRX1 and both together. Now in all the cases, whether it is PRX1 alone, DAF1 alone, or DAF1 plus PRX1, actually it can protect the strain against hypersalinity, oxidative stress, PEG is dehydration. So conclusion is that DAF1 actually interacts with PRX1 and it has no interacting partner whatsoever which is there in the chloroplast. It's a full proof that DAF1 help plants or promote resiliency in plant only by its intimate partner, that is peroxyredoxin, which is there in the mitochondria itself, and the entire pathway has nothing to do with chloroplast. I believe that I am already almost done with this lecture. So finally I will tell you that we draw a working model for this. The model is this. Identified a protein by means of proteomic analysis and through organelle proteomics. The protein name is DAF1. We also identified its interacting partner, which is PRX. PRX having two forms. One is PRX 1.1 and PRX 1.2. And we picked up 1.1. We introduced both these in rice, as well as we knocked out the DAF1 from the plant. And the result is summarized here. When plant undergoes stress condition, it is a powerhouse. But when plant undergoes stress condition, it produces more and more reactive oxygen species, ROS. And all of you know, the more the production of ROS, it is actually very harmful for cell and the organism. So in one way, this is the pathway. Alternately, under dehydration, the plant, within the plant, a DAF1 protein induced. And when DAF1 is induced, it activates the function of PRX 1.1. And actually this protein also redox protein. Now we believe this also redox protein. Now this one redox protein with another redox protein, it makes a redox complex. When this complex is active, then it accelerates the catabolism of ROS. The moment it is removed, then the mitochondria goes back to normal function. And once the mitochondria goes to normal function, it helps plant surviving a stress condition. I think I am able to give you some sense that how a laboratory can start with a journey from protein to cloning, getting partner, and then put into the native system or kick out that gene from the system, and can establish a story that how one protein can be characterized by different mean and come to a conclusion that how beautiful this protein is. So with this, I say goodbye to all of you. Before that, yeah. So I have a group. I give lecture only. I don't do work in table. So this is Deepak Gayan, who is the main worker in this group, and helped by Pragya Borua and Nilesh Lande. All their names are there, and I get money from Department of Biotechnology CSIR, but I get more money from my own institute. And lastly, actually, I have a relation with Jamia Hamdan. 10 years back or so, a beautiful student came to my lab. She wanted to do PhD with me. I took her. And another story is there that how do we identify a cell wall protein which can promote the stress tolerance in plant, and take it from me. That paper we published in a very good journal, as well as that gene got an US patent. Now Shaista Parveen is a faculty in Saudi Arabia. Thank you very much. Thank you, Chairman.
Thank you, Dr. Chakravarti, for a wonderful presentation. And I think you can take 2 questions. Come on, yeah.
It was not at all a plenary lecture. It was a lecture for you only, so you should ask questions.
And introduce yourself. Anybody ask a question, just introduce name and department, that's all.
Sir, excellent question. Actually, I'm Poonam Ray from Regional Center for Biotechnology.
RCB.
Yes.
Okay.
Sir, excellent work. I, I will have one question only.
Yeah.
You took, rather than taking native promoter, you overexpressed the protein to check the, the function of the protein. I would like to know why, because overexpression most of the cases it gives us, because you are checking the function in a scenario where you are adding stress to the plant indirectly. So you could have used the this native promoter to check exactly why, what is the actual function of the protein in its native.
I got your question, but I make a correction. Actually, you can put a constitutive promoter, native promoter, or any promoter. The problem will be whether the plant is under stress condition or unstressed condition, the protein will synthesize, because it is normal constitutive promoter. I believe, I don't remember exactly if it was written in the slide, I believe we use RB29, a promoter. RB29 is a promoter. The beauty of this is that unless there is stress condition, that one will not, bulk amount of protein will never be produced. The moment the plant sense stress condition, then RD29 only will induce the protein to produce. Otherwise, what is the need? You do not put a protein to constitutively express the protein all the time, 24/7. You give this a rescue route that whenever plant will undergo stress condition, then RD29 will help the protein to express at higher level.
That's what I got confused, because it is overly expressed. So even if the stress is not there, it will be expressing, right?
Very minimal level, subtle level, because it is not inducible promoter. You always stress responsive protein you put under stress inducible promoter. because whenever it is not needed to produce that protein, forget it.
Thank you. Any other question? One more question. Mike.
I am audible, but
Yeah, but
rest of
others
the
are not.
people may not. You are audible. Yeah.
I just want to know what made you choose that hypothetical protein. Like I said, I missed that part because there were many genes that were showing the same expression, differential expressions, but you still chose that hypothetical protein. You could have worked on any other protein.
I could have. Why did I?
But yeah.
I think a little bit I told you myself mistake. Look, you got a SOD. SOD also induced. In that 101 list, SOD was also there. So people will think that it is a stress condition, oxidative stress. So you put SOD back into that, so it will produce more SOD and it will actually accelerate the catabolism of process. But it is a known function. The beauty of Nobel thesis and Nobel paper is you take a protein which nobody knows the function. You give a name of that protein. That I have worked out with a protein which does it. Why in the if go to PFAM database, then you will find 2700 protein is still there which doesn't have any functional property, unknown function. But why God has given this protein into the system? It has a function yet to be identified. That is why every PIS laboratory, two types of students are there. One student will tell ATP synthesis you give me, I will clone it and overexpress. Another student will be there who will always will tell I will pick up an uncharacterized protein and characterize it. That is the beauty.
So that was your motive to do this study with.
Obviously.
And in a situation where out of 111 proteins, you like there was a chance that there was no hypothetical protein, and all the proteins were annotated.
Many others are there. That is the reason the list of paper I have published, how many organelles are there? Cell wall, cytoplasm, mitochondria, nucleus, etc. etc. I published more than 100 papers. From where this protein came? In one student you give one uncharacterized protein. Another student you give another uncharacterized protein. Another student you give another uncharacterized protein. Let every student come with a new story. That I have taken an uncharacterized protein and this is the function of that protein. That is the reason.
Thank you, Dr. Chakravarti. Last question, please.
Hello, my name is Prashant from RCB. So it's an excellent presentation. It was very clear. So the ROS which was there, is it organelle specific? For example, it was from mitochondria.
Zinc SOD, copper SOD, these are established pathway are known. Which SOD works in which organ. Yeah.
So our next batsman is Professor Mishra from IIT Delhi, and Mr. Mishra will talk about mitochondria. Please come and introduce yourself and then talk.
So by the time he is starting the presentation, let me thank the organizer for calling me. And yeah, so am I audible? Hopefully yes. Okay, so I would like to thank the organizers for inviting me, especially Professor Abdi. And thank the chair for inviting me to give the talk. Now today, I'm going to take away from you, from the plants to the from plants to your the bacteria, and are you able to?
So, 1 minute.
So here, all of you know that there is another global challenge which is appearing and is increasing, is antimicrobial resistance. And I'm going to talk about that, how we can address this global challenge. So my title of our talk is antibiotic stewardship and how one can, address the global challenge during and has to be sustainable. The topic itself talks about stewardship. That means you are not going to use antibiotic unless it is required. So on one hand, you are going to help helping the human health, but on the other hand, you are not contaminating the environment. And here, there is a concept which has appeared is that we are looking at the global challenge. And this global challenge is not just for the human health, it is for the health of the environment, health of a plant, health of other, So why we need antibiotic stewardship? As you can see that WHO in July 2020 has shown that there is a biggest threat which is appearing because of the antibiotic resistance. And it is not going only to health problem, it is also going to address, we're going to talk about food security and as well as sustainable development. So what is the antibiotic resistance? Most of you know that many of the bacteria, they are becoming resistant to antibiotic, and that is why when you use any antibiotic, they are not, being killed. This antibiotic resistance can happen to anyone, at any age, in any country. So it is not, constrained to one country. It is a global challenge. And what you find is that though antibiotic resistance is a natural process, but it is happening, it is advancing because of misuse of antibiotic. It is not only that we are taking antibiotic, it is going to be given in poultry, it is given in aquaculture, and ultimately it is going in the environment. And what is ultimately happening? It is happening where the new infections or any infection which can be treated very easily, like pneumonia, like tuberculosis, like salmonellosis. All of them, if your bacteria become resistant, you can't treat them. It is going to lead to the mortality. And that is where we have to address these issues. Now, antibiotic resistance, many of the therapies, they are not going to be effective against this drug resistance bacteria. There is there are various pathogens which are coming under escape category. They are, for example, Enterococcus, Staphylococcus, Klebsiella, then Acinetobacter, Pseudomonas, Enterobacter. All of them, they are becoming resistant to antibiotic. Now, question is that how we address it and what is going to be challenged. Now, most of us know that a huge population, I'm just taking data from US, something like 23,000 people, they are dying because of antibiotic resistance appearing in their area. But there is a challenge. If we don't treat or we don't solve this problem by 2050, something like 10 million people, they are going to be affected, they are going to die because of this death. See, what is the challenge? In Corona, we had how many? So far, how many people they died because of Corona? Approximately 7 million. Here, 10 million every year people they are going to die if antibiotic resistance becomes, a case. And now if that happens, we are entering post-antibiotic era because before antibiotic was developed, people were dying. Before village was, dying because of one kind of infection. Now, if it becomes resistant, that is the post-antibiotic era where we have a big challenge how to tackle that. So there are various ways one can reduce the antibiotic consumption so that antibiotic, is not becoming resistant, bacteria is not becoming resistant. One can also increase the efficiency of antibiotic. Over years, you find that there are not many antibiotics which have been developed. One has to also look at the accurate diagnosis. Today, you go to the doctor, you say that I am having some symptom because symptom is similar to viral infection, he will give you antibiotic. So what you need is that you need accurate and early detection. And of course, we need alternative treatments like antimicrobial, which can be as such that which can target many places inside the bacteria so that it can be killed efficiently. There are methods, for example, biochemical tests, ELISA, PCR. They're very well developed methods, but the problem is it is time consuming. There are issues of sensitivity. Some cases it could be sensitive, but there are many challenges. Suppose there's limitation, and if you are using a particular probe, that may become a challenge. But there are complicated procedures, and most important is that you need a manpower who understands everything, does the experiment, and gets the data. What we need is that we need something which is a real time, which is easy, it is rapid and highly sensitive. And of course, you don't need the manpower. You just put the sample and you will be able to detect it whether yes or no. You can see the indicator and that can tell you that whether this is happening. So here we are going to talk about a new technology where we are trying to develop biosensors, and these biosensors are basically analytical device. So I'll be talking about something which is interdisciplinary, where we are talking about physicists, chemists, as well as electronic engineer, electric person, as well as biologists. They are required. So it's a multidisciplinary where you need all kind of support to develop these technologies. And here, what you have is typically for students, there is a target analyte, you have a recognition element, you have a transduction, and then signal processing, and finally you get the signal. Now, a typical sensor, biosensor, it is not necessary that it is going to detect something which is present in biological system. It can also detect something which is present in the environment. But what you need is that biomolecular interaction. That is what is important. And now the challenge is that can we detect things in non-invasive samples? So, for example, you can detect things in saliva, you can detect in the sweat, you can detect in the urine sample, rather than just going for blood where you are taking in, which is invasive sample. But when you are developing biosensor, what you are interested in, you need to understand about limit of detection, you need to know about the sensitivity, you need to have selectivity, as well as you need that the sensor should be reproducible, and then, of course, you can miniaturize them, and that is where the challenge comes. So we are working in our group where we are developing different biosensing platforms. These are different platforms based on their electronic characteristics. We are working on impedance sensor, FET sensor, field effect transistors, surface enhanced Raman spectroscopy, photonic crystal-based sensors. So all these sensors we are working not alone with the physicists, with the electronic people, with others who are developing the material and material the device. And what we are doing is that we are focusing on the receptors, the bioreceptors. So we are targeting how one can develop these bioreceptors, which are a small molecule, could be antibodies, could be aptamers, could be MIPs, and that is where our challenge comes. And doing so, we could analyze different bacterial toxins, cancer biomarkers, various other diseases like liver disease, etc. Okay. Yeah. So now I will skip it because we have already discussed that there is one challenge. The material scientists, they are trying to develop new material which can be useful for developing sensors. So you need certain important things in the material. For example, response time should be fast, there should be dynamic range, there should be limit of detection, signal to noise ratio should be high, as well as there should be specificity. And here there are a lot of nanomaterials which are being developed. Now, in biosensing, I have already talked about, so I'll skip this slide. This is something which we have tried and worked with, a very simple antibiotic. Now, people they have worked with antibodies, we work with antibiotic. This antibiotic is a small molecule. It works with the hydrogen bonding, which is known based on the gram-positive bacterial surface, and it has a very high binding affinity. Now, because of this, we developed a device which is a highly sensitive and selective device. You have silicon oxide, then WO3. On the WO3, we have immobilized this antibiotic, and then this antibiotic was specific for gram-positive bacteria. So you can see the binding with the gram-positive bacteria. Now, the reason why we have selected is because of the material is because of the porous morphology, conformation, and high degree of transfer rate, so that your sensor, when it's developed, you can have very good efficiency. So this is the structure of sensor, which can show you that the actual data, that how the sensor they bind with the bacteria. And here we have talked about and discussed about the SRS, which is one of the well-known pathogens. Now, once you develop the sensor, you do a lot of characterization. You go for the data where you understand what is the the material characteristic, whether it is immobilized. You go for FTIR, you do FESM, you do atomic force microscopy and scanning electron microscopy to understand that exactly bacteria is a binder. And after you do that, you do electrical characterization. I will not talk too much about the electrical characterization, but it's important that you can differentiate. You can see the data. Different concentration gives a different output, and that is what is based on well-known plots known as the Bode-Nichols plot, and you monitor the phase change. The important thing is that dead and live bacteria can be differentiated by monitoring with the electrical data. Then you can determine the limit of detection. You can see up to 20 about the 2 bacteria, 100 bacteria per ml can be detected. That means if you are taking few microliter, single bacteria can be detected in this. And on the right hand side, you can see the graph. Because of the presence of vancomycin, only gram-positive bacteria gets a signal, gram-negative bacteria doesn't show the data. And that is why you can discriminate between gram-positive and gram-negative bacteria. We did a lot of other characterization where you can differentiate. You can do this in presence of different physiological parameters, where you can do it in a pH, you can do it in presence of plasma, you can do it in other solutions, basically FBS. Now, once we did it with impedance, we have moved on to another technology, which is a field effect transistor-based technology. Here, as you can see, we have silicon oxide, MOS2, and TiO2. Here I have shown that how the hybrid material, one is MOS2, another is the TiO2, their hybrid characteristic makes a difference. Otherwise, biologically, we have put the same molecule, same basis for their characterization. You can see that how these bacteria, they're present, and there's a difference between gram-positive and gram-negative bacteria. So both of them, they're same, but here, the idea of doing field effect transistor was that it is CMOS compatible technology. It is technology which is similar to any other technology which is used for developing semiconductor material or semiconductors, which is present in our mobile or in other devices. Okay, this data is basically characterization data showing how is the FET-based data is showing their binding. This is the AFM data showing the MOS2 and TiO2 channels. One can discriminate them. These are some further physical characterization. I'll just move on to the one. Yeah, so here one can do online monitoring. You can look at the characterization, MOS2 and TiO2 FET, that gives you online where you can monitor with the time. So it has a compatibility where you can put it with a device which is, point of care device. In this, we have shown what is the limit of detection. Something like 50 CFU per ml can be detected. And on the right-hand side, it shows the discrimination and sensitivity of gram-positive and gram-negative bacteria. This data shows that, yes, what is the output characteristic and transfer characteristic, especially it is of interest for electronics people who are interested in how good material is. We have demonstrated that how this data can be exploited with different concentration of the bacteria. Now, this is the one where I'm interested in. On the right-hand side, you can see more or less all the data shows same sensitivity. Whereas on the left-hand side, you can say the difference between gram-positive and gram-negative bacteria, as well as different types of material. When we take just TiO2 or just MOS2, you have a lesser sensitivity compared to when you have hybrid material. So it is important that one should have nanomaterial, which is a hybrid nanomaterial, which can give you the data. In addition to this, we have also done something where very simple technology which has been used. Here, you know that most of the bacteria they produce H2S in a very, very low concentration. So we developed a method using Raman spectroscopy there, and in addition to Raman, here we have demonstrated that how very low concentration of H2S can be measured using silver nanorods. And these silver nanorods were developed using a method which is known as a glancing angle deposition method. That means you are drawing the nanorod physically, and this nanorod is having an angle. So because of the presence of angle, the hot spot developed is much better, and it can show you that how they can be differentiating the live and dead bacteria. And not only that, it can also look at the sensitivity of the data against different, bacteria, whether they are antibiotic resistant or sensitive. This was also coupled with their other than that, we have also seen that what is the contact angle, because these bacteria, when they're live or dead, their contact angle is found to be different. And this is very clear that one can show that ampicillin resistant and ampicillin sensitive strains, they will have different, characteristic value. And you can, compare with the classical method of fluorescent sensing. And once you can couple it with the, you can take because it is based on the color, you can connect it to the mobile, and your mobile can sniff the presence of the bacteria. Then we have moved on to SIRS, which is known as the surface enhanced Raman spectroscopy. I'll not go into too much of basics, but yes, it is enhancing. So whatever the Raman data you get, if I put some plasmonic material like silver, it will enhance the data, and the data enhancement could be up to the 10 to the power 7 or so. That means those data, which is almost absent, can be monitored. So we have developed a method by which putting the PDMS, one of the material, one can detect these bacteria, and you can enhance the sensitivity of these material by several fold in terms of 10 to the power 7 or 10 to the power 6 fold. So we had a question whether we can use this data for measuring viruses. Yes, we do it. We have used, and in fact, we have developed a device which is like size of the mobile. And based on that, you can detect viruses. And here, we have used with the clinical data which was found from Malaria Research Institute for detecting dengue viruses. We have also used another technology which was developed with the physics group. Here we have developed the technology which is based on guided mode resonance. And now this technology we have sold to a company in Bombay, and we are developing different types of sensing for material, could be biomaterial or could be bacteria. Okay, I'm sorry, it is. Okay, so now the question comes that where the biology comes over here, because a lot of physics we have talked about, a lot of chemistry we have talked about, immobilization we have talked about. Here the major thing is that we need to have a receptor which is a bioreceptor which will be specific. And now for that, what we did is that we did molecular imprinting. Molecular imprinting is a very simple technology where what you do is that you take the material. Here we have worked with a magnetic material. What you do is that on this magnetic material, you have a template, and this template will be allowed to interact with the polymer. After polymer interacts, you remove the template, the impression or imprint remains. Now this imprint can allow the binding of the same material, which is a template material. So you can take a small molecule, you can take viruses, you can take epitope, whatever it is, you can use them for developing MIPs or molecularly imprinted polymers. These are known as the synthetic antibodies. They can be produced in the larger scale, and that is why it is important. Not only it can be very robust compared to the antibodies, it can also be used at harsh condition because it's not protein. On the other hand, you can also see that if the cost of antibody is $100, you can produce it in $1. So there is a cost effectiveness. So there are many companies which are developing these molecularly imprinted polymers. Here, I'll just move on to the, this is showing how this template, here we have used pyocynin, one of the toxin produced by the bacteria, and imprinted it. And once you make the imprint, because it is on the magnetic material, you can separate the toxin. So the interest was that you can use the biomarker, which is like a toxin that is produced by one of the common bacteria, and then you can separate it out. Once you separate it out, you can demonstrate the presence of bacteria. So you are showing the presence of bacteria even in the absence of actual bacteria. You are looking at the toxin. And it is important that because many of the infections, it is not important that the bacteria is present, you need to look at the toxins. We have similarly developed for another bacteria, Staphylococcus aureus, where we have imprinted protein A, which is another marker for this bacteria. Now, while developing this, it's very important that you should develop a rational design. You do the molecular docking, you see how the monomer interacts with these molecules, and once you know the interaction, then you can develop a new kind of a material which could have better binding affinity. Here we did both monomolecular as well as the multimolecular docking and demonstrated that yes, it is possible that out of thousands of the combination, you can select few which can have the best binding. So here, this is based on silicon-based monomers. You can do the imprinting, and based on that, you see the data. Actually, we normally believe that the lower the maximum value of delta minus delta G is the one which will show you the best binding. But our results were different. And the reason why it was different because there were multi places the binding was occurring. So it is not only single monomer, it is multi-monomer docking that helps in the deciding their ability to bind. Now we have also moved on to epitope binding. Because proteins are a big molecule, if you develop the MIPs, they slip it out. So what we did is that we took the epitope, we characterized the epitope, and based on the epitope, we did the binding affinity. And what we observed is that these epitopes, so here the work was done with SARS-CoV-2 protein, and on the lower side you can see the blue one, which is showing that actually when we take VLP, virus-like particles, not viruses, they show very high binding affinity. So there are many things which are being developed based on molecular imprinting, and there are many sensors, many diagnostics, in vitro diagnostics which are being developed. And not only that, once you develop this technology, now question is that how to treat them. We developed some of the material, which is a combination of the material of silver or other nanomaterial along with the bioactive molecule, which can be used as an antibacterial. So there is a couple of data which shows that how combination works. Other than that, we have also looked at different combination of antibacterial with antibiotic, as well as we have looked at the biologically active molecule, which can show their efficacy. Very important thing is that when we are developing these new antibiotic, it may be very good, but if it kills mammalian cell, then it's of no use. So what we did is that we looked at the selectivity, how selective they are vis-a-vis means the concentration which kills the bacteria and the concentration which kills the mammalian cell, how they are comparable. Another technique which was developed with MU, where we have treated with a diabetic foot ulcer. Here we developed a photodynamic therapy where you take nanomaterial, put along with the dye, and then you excite them with the photons, and then you kill them. We have also developed different types of broad spectrum inhibitor where antibiotic resistance develops. Now with this, I have done with my first part. How much time? Okay. So now I'm moving on to another thing which shows about the sustainability. Now, when we look at the sustainability, we all know that the electronics are growing very fast, and each one of us are having several mobiles. And what is happening? These electronics, when they are coming as a waste, they are going to be a huge challenge how to treat those waste. What we try to do is that, being a biochemist, I started working on proteins. If these proteins can be used as a material which can substitute some of the semiconductor material, and we can employ them for developing memory devices, or for developing a sensor, or for developing something which is commonly used in the electronics using silicon. So we are working on non-silicon based technology, and here we have isolated a very simple protein, which is the S-layer protein. It's found in most of the bacteria, archaebacteria is present. They are very well organized, and based on that, we have developed something which can be used as memory device. So memory can be, where we have HRS and LRS, there is a huge difference, and has a very high capability to be used as memory devices. Now we are working, I'm not showing the data, but we are working on some proteins which can be used as a neuromorphic computing, where you are mimicking our brain, the proteins which are present in the brain. Look at the brain, how much less energy it requires to store the data compared to the computer. So what we are trying to do is that, can we develop those proteins which can be used for developing memory devices? In this line, we have developed metalloprotein-based field effect transistors, which can be showing the enhanced switching behavior. So here we have taken very interesting protein which is present in Pseudomonas aeruginosa, isolated them, cloned them, expressed them, and then used them for developing as a part of the switching behavior of field effective transistors. These are the basic data. On the lower hand side, you can see the optical image of the device where you have SEM. On the right hand side, you can see the device where you have a source and drain, and in between you have the protein, which is adjuvant protein, and it can show you the characteristic shown in basically field effect transistors. We have also used for ultraviolet detection, and with this, I will come to the end of my talk. I was lucky to get the funding from MIT. MIT is the Ministry of Electronics and Information Technology. It has given 2 times funding, so we have a lab, which is a nanofabrication lab. Twice we got 50 crores, then we got another 7 crores. And we are running this with the help of other institutions like DST, which is giving 20-30 crores every 3 to 5 years. We have got grants from DST, ICMR, DBT, and more interestingly, from Lockheed Martin, US company which is making fighter planes. Sometime back, they were interested in learning something or doing something in nanotechnology, so they funded us. So with this, I would like to also thank my collaborators who are from physics department, electronics department, as well as different international groups, for example, from Germany, from Norway, from Australia, as well as we have a funding from and collaboration with US. With this, and a lot of my PhD students and those who have participated in this as a project or as a PDF. Thank you very much.
Thank you, Dr. Krishna. Very important topic where you have spoken a lot, and very significant work you have been doing. Recently, I was the CEO of POIT Innovation Competition at just designed a couple of weeks ago. And we had a project of 190 crore rupees, 70 crore from government of Rajasthan, 130 crore from government of. And they are also working on senior lines, semiconductors, they are working on semiconductors. They are also supposed to sterilize sepsis and antibiotics. So I would suggest that we can try to collaborate with them also. And very, very significant work. And the alternative approach is when you are using the protein and chemical. That is also very important. And I think I will say some of the students here, because we have been talking that antibiotic resistance is one of the biggest challenges of the coming century. But honestly, if you ask me, we are not doing enough to address this. I have been in ICMR for 36 years. I was 10 years in food authority, dealing with the vendor exercise restriction antibiotic. And we can talk much about that. Recently, ICMR has one task force antibiotic. And also you mentioned about the antibiotics which we are taking protein correction. That is also very important. We cannot address that issue. What is we are taking, what doctor is giving to us, other thing also like depending on the diet. Where indiscriminately antibiotics are used by the farmers. It's a very important area, so I will urge some of you that you take up this topic, and there is a lot need to be done in this area. So thank you very much once again.
Thank you. In fact, in Jodhpur IIT, one of my collaboratory whose data is presented on electronics, he's a faculty member in electronics.
Right now, Ajay Agarwal is doing.
Yeah, yeah.
And this project is a genetic Professor Shantanu Joshi, who is
He is colleague of
mine. So he did last. You are representing that, and you can talk to him also. Anyway, so I think before I give this mic to Professor Chaturvedi to conclude, I would like to thank you, all three speakers once again. Dr. Chaturvedi, for wonderful presentation, Dr. Bhattacharya, Dr. Mishra, all three presentations were very nice. Thank you very much to all of you. And also I would like to thank the audience here. Wonderful audience. And I observe everybody, most of the audience was very alert. They listened all the presentation very well. So I think round of applause for all our audience. And also, I would thank my co-chair, wonderful person, who will conclude. And I'm enjoying this computer company. And also, I would like to thank all the majors, Mira, Lisa, and myself, everybody, for giving this opportunity. Thank you very much. Sir.
Thank you very much, sir. Actually, I don't know how to conclude the session. I have two questions to Professor Mishra.
Yeah, sure.
If I missed this, the last part of this talk you said protein, brain protein, and identified as a bacteria.
Yes.
So what the brain protein does in bacteria?
So I'm not talking about the brain protein and bacteria. What I'm talking about is that in a brain we know that the proteins are present and their function is where they store a lot of memory. And the amount of energy required for that is very, very low. So can we mimic that scenario and we develop neuromorphic computer type system using proteins? So what we are trying to identify is, and some work is going to be published very soon, where we are going to use these proteins, and they are the ones which are requiring much lesser energy, and about that 5 times lesser energy than the energy required by the semiconductor material. So question is that can we use that? Can we have sustainable development where there is no problem of degradation of those electronic waste on one hand? On the other hand, can we put them inside our body and they are automatically degraded and there is no complication?
Now I have a lighter, it's not question, simply. You have crores of rupees. Why don't you give some money to poor consultants?
So we are developing a free training program across the India where anybody can come. We pay for them to stay and learn the semiconductor technology. Yeah, anybody can come and learn, and that is open. So it is called as an INUT program in Indian National User Program, where you can go to our website, and it is conducted by 5 IITs and ISC. 6 of us, we are there together, and anybody is open to get a project, basic training, etc.
Thank you very much, sir. I am privileged to speak with you and sharing a session. And I echo with you. It's a wonderful audience, and I really enjoyed in front of you sharing my data. And I wish you all the best for new PhD, who are in masters. I believe that if I have give you some knowledge about protein biochemistry, it is a great thing for me. Thank you very much.
So as we conclude this session, so it's time for felicitations. So may I request the chairpersons to felicitate the speakers. So as Professor Chakravarti was also a speaker, so may you please felicitate Professor Chakravarti. So may I please invite Professor Prashant Mishra to be on dais and the chairpersons to felicitate Professor Mishra. Good. So may I now please request what the organizing secretary, Professor MZ Abdi, to be here on dais to felicitate the chairpersons. Yeah. So this is the Professor Tureja. And for Professor Chakraborty. So thank you, sir.
Now I would like to request the chairperson to present the memento to the moderator, Dr. Jalu. Now I invite all the speakers, chairperson, moderator for a group photograph, please. Group photograph. Thank you so much. Thank you so much, sir, for the wonderful session. Now it's time for the tea break. I would like to request all our guests to move towards the lobby area for tea. After the small tea break of 15 minutes, we will resume.
अनम्यूट है सर, म्यूट कर देना चाहिए ना सर, अनम्यूटिंग सर, लाइव यूट्यूब पे भी सर। पसंद नहीं हुआ
Okay, so good evening, everyone. So let's resume the session with renewed energy after the tea. We'll now begin the technical session 1A, and the theme of the session is agricultural innovations and environmental sustainability. I would now request Dr. Javed Ahmed, Assistant Professor in the Department of Biotechnology, to moderate this session.
Thank you, Madhya. Good evening, respected guests, speakers, and participants. Welcome to this technical session. It is my honor to introduce our distinguished chairpersons for this session. Dr. Ajay Arora, sir. Welcome, sir. Please join us. Dr. Ajay Arora is a principal scientist in the Division of Plant Physiology, ICR-IRI, with extensive contribution to plant molecular biology and stress tolerance research. His work focuses on improving crop resilience through advanced physiological and molecular approaches. He has been recognized with prestigious awards, including Nanaji Deshmukh ICR Award, J.J. Chenoy Gold Medal Award, and AAS Award by Indian Society for Plant Physiology. He has served as editor-in-chief of Indian Journal of Plant Physiology and a member of the Research Advisory Committee at ICR-IIOR Hyderabad. Now, our other chairperson is Dr. M. Iqbal Raza Khan. He is GM of our SCLS Jamia University. Now, Dr. M. Iqbal R. Khan is a distinguished scientist specializing in plant stress physiology, environmental sustainability, and crop resilience. He is assistant professor and Jamia Hamdard and also serves as a deputy director at the Center of the Environmental and Sustainable Development, leading initiatives in environmental conservation and sustainable agriculture. His impact extends globally through his adjunct position, first adjunct associate professor at the University of Western Australia, collaborating on agriculture sustainability research, and other is adjunct professor at the Department of Plant Biotechnology, Korea University, South Korea, contributing to advancement in plant biotechnology. His extensive research portfolio includes contributions to plant stress biology and crop resilience, making him to a key figure in advancing sustainable agriculture practices and environmental research. Thank you, sir. May I request to our chairpersons to introduce our speakers of this session. And please, conduction of this session.
Thank you very much, Dr. Javed, for very nice and beautiful introduction, which nobody does for us when we speak. Only during the conferences, only we can. Even not at home also. So thank you, Dr. Malik Zainul Abid, my senior, who brought me to this particular place because I'm a regular visitor and maybe I don't know how many of you recognize me. I used to come for many biology, many practical exams and many lot of plethora of exercises. So nice meeting all of you. I know you must have been tired by now. Since morning people are listening to many lectures. But this was a very good welcome tea break. So maybe little bit of refreshment you got it. Hope so. So I am thankful to my young budding biotechnologist, Dr. Iqbal, with me. So what I will do, shall we start with the series or shall I deliver the last? Or this is up to you, Dr. Iqbal. Series? The series on okay. Then I will start. Okay, thank you.
स्लाइड इससे हो जाएगा जैसे इससे हो जाएगा एंड there was a pointer so I'll get to the pointer ये क्या है so इससे forward and इससे backward just so उसे नहीं कुछ नहीं टच नहीं करना yes
Okay. So Dr. Iqbal, please note down my times. It is strictly 20 minutes. 2 minutes before 20, maybe after 18 minutes, give me a signal.
so also this as a pointer I'll keep it here
Okay, no need. So, I am Dr. Ajay Arora, Principal Scientist in Division of Plant Physiology at ICAR-IARI, Indian Agricultural Research Institute, New Delhi. Right. I know most of you are students there. I think more than 95% sitting there. And that too, out of 95%, I think 80% are sitting by force, I suppose. If the opportunity will be there, तब भाग जाएंगे. I understand that. But सुनना तो पड़ेगा, है ना? हमारी कोई सुनता नहीं है देखो. So this is the topic which I have chosen because right now most of my students from the last 1 decade are working on this particular area. Stay green. अच्छा लगता है सुन के. Stay green, like stay ever young, है ना? Who wants to live longer? How long? Sorry? 100 years. Very good. One of the student I got who wants to live more than 100 years. Very good. Most of all the students, they don't want to live more than 60 years. I don't know why. Okay, anybody? And why you want to live more than 100 years? Very good. खुश किया यार आपने. I'm so happy. Yeah, exactly. So there must be you have heard mostly about many groups in the world. They are working for human beings. How they want to become younger for a longer period, and that too disease-free, right? We wish we can develop our own strategy, right, for betterment of our younger look or younger disease-free life. But plants are bechara. Bechara in that sense, because why I'm using bechara, they are sessile in nature. They won't be able to roam around. They cannot run around. You can exercise yourself, plant can't. They have to maintain or they have to sustain their own kind of strategy to become younger, right? So that's the reason I chose this particular topic: stay green, staying alive or going to die during terminal senescence. I know if I'll go into nitty-gritty of all the research which our group performed in the last, say, 6, 7, 8 years, so it will be difficult to understand because this is an exposition of kind which you should get an idea what we people are doing. That is most important. I'm not going to detail of everything, but you should understand how to become young or how this stay green type of plants should be like that. So to identify which particular, I'm working in the case of wheat. Wheat, to understand, is very, very important and staple food crop in our country, right? So you can discuss later also. I understand you must be tired. I understand. So try to listen. I will make it very simple because I know since morning lot of bouncers. Bouncers, you understand? हाँ, क्रिकेट में होता है ना, ऊपर से जाती है बॉल. मेरे को मालूम है कि जो भी मैं अभी बोलूंगा वो सब बाउंसर की तरफ जाएंगे. because we are having expertise in our area. But for you, you are listening maybe 8 to 10 lectures since morning. So कभी इधर की बात सुनो, कभी जीन, कभी प्रोटियोमिक्स, तो ऊपर से जाए. So इसलिए I'll make my talk very simple, by which you should understand what is the strategy. So there are two kind of system in the plants. One is photosynthesis. You understand photosynthesis? Everybody understand, right? So one is current photosynthesis, by which the younger leaves can make the carbohydrates. Okay, but during stresses, stress is again a problem. Why it is a problem? Plant can't differentiate between different kind of stress. If drought will be there, they have to cope up with this. If heat is there, they have to cope up with that. And if salinity is there, they have again to cope up. And there are so many kind of abiotic stresses, right? And other part is biotic stresses also. So very difficult to sustain with these kind of stresses. So plant can't differentiate between these kind of stresses. But what me and my group thought about to start working with drought and heat, combined stress. So because in the case of wheat, these two stresses are very, very cumbersome for the plant to survive. So say the yield is, if 100 tons per hectare, I am giving you an example, 100 tons per hectare. And if stresses will be there, both the stresses, it will be less than 50 tons. So how to increase or how to enhance the yield level or the productivity of wheat crop? So we thought in that way, what to do under stress condition. One is photosynthesis will be suffered, right? So there will be another terminology. One is staging, another terminology is SRM, stem reserve mobilization. So how this stored material, आप भी करते हैं ना, कई बार पैसा नहीं होता है तो बैंक में रख लेते हैं, तो बैंक से निकाल के फिर यूज करते हैं, ऐसा होता है. So इसमें भी ऐसा है. So जब current photosynthesis is not able to help, because वहां तो है नहीं, because abiotic stresses में तो ये सफर करेगा, plant will suffer, है ना? So in those conditions, जो stem में stored होता है material, that will be carbohydrate stem reserve mobilization. That carbohydrate will be helpful for the developing inflorescence of the developing pea. So simple सा phenomena है ना? But क्या होता है? How to identify these cultivar genotype germplasm which is showing better performance under these kind of abiotic stresses. अब आपके सारे बच्चों को, maybe 100 students are there, अगर आप सबको 1 किलोमीटर रनिंग करा दी जाए, कुछ तो नंबर 1 पे आएंगे, कुछ नंबर 2 पे आएंगे, और कुछ वो जो 5 मिनट में कर जाएंगे, कुछ होंगे 15 मिनट के बाद पहुंचेंगे, कुछ 10 मिनट बाद पहुंचेंगे, right? So we have to identify the potential or the variability in those kind of germplasm which we have. So we did these kind of exercise, number 1. So which is that particular genotype or germplasm which is showing the better performance under these kind of abiotic stresses in terms of staging rate. Staging rate, if I am talking about, that means the photosynthesis, the functional photosynthesis. And the second is stem reserve mobilization. But दोनों को साथ में लाना है. We have to come across with the both kind of traits together. Very difficult, I tell you. It is very difficult. But during the evolutionary process, there might be a possibility that these two traits combine together through recombination. That is our answer. That's what we thought. And we tried to identify through germplasm, through genotype, and through mapping population. If I am using this word, that means real population. recombinant inbred lines. So through real population and through germplasm or through genotypes, we identified few germplasm or few rays which is showing better performance under these kind of abiotic stresses. Simple, very simple. Okay, but both these traits, mind it, focus on this particular point. Both these traits, stay as well as the stem result mobilization, both are opposite to each other. ये काम साथ साथ नहीं चलते. So जिसमें SRM ज्यादा होगा उसमें स्टेग्रीन या फोटोसिंथेसिस रेट कम होगा, जिसमें फोटोसिंथेसिस रेट ज्यादा होगा उसमें SRM कम होगा. So we have to identify those kind of system or those kind of real population or mapping population which is showing these two kind of states. That's it. That's what I wanted to convey. मैं थोड़ा बहुत आपको इधर-उधर के evidences और ये वो सब मैं दिखा देता हूँ, है ना? What we did, we identified certain kind of QTL also. QTL तो सुना होगा word? Yes. Yes. Say yes. Yes. Thank you. So we identified certain QTLs, certain genes, which is associated with these two kind of processes or traits, and how we can enhance the productivity, yield level, or the production in particular our crop variety. So from the last 7 to 8 years, we did these kind of experimentation through various kind of system, right? So have you heard about certain kind of plant hormones also? नाम सुने हैं किसी ने? सुने हैं? कौन-कौन से हार्मोन? Very good, smart बच्चे, very good, बहुत बढ़िया. अरे यार आप सबको जानते हैं, मेरे को बताने का जरूरत नहीं. So these 5 which you told me are the classical hormones, plant hormones. Apart from this 5 hormones, auxin, gibberellin, cytokinin, abscisic acid, ethylene. Okay, apart from this, there are long queue of another plant hormone which wants to come into the bandwagon of plant hormone category, right? Which we used to call them as plant growth regulators. और latest बहुत सारे आ रहे हैं. Melatonin, सुना है कभी ना? है ना? सुना है? Happiness hormone, जो ज्यादा हंसते हैं ना, उनके अंदर मेलाटोनिन बहुत है. उनको कभी बोलना नहीं है कुछ भी, उनसे सीखना है. उनको है ना, बताना है कि भैया आप हंसोगे तो हम भी हंसेंगे, लेकिन उसमें भान नहीं है, ये याद रखना है. ठीक है? So these kind of hormones are helping in these kind of two traits. That's what I wanted to tell you. तो वो क्या है? Cytokinin, which is associated directly with plant senescence process, है ना? जिसमें senescence ज्यादा होगा उसमें cytokinin कम होगा. सीधी सी बात, है ना? And you want to say something? Please, please. अरे इसको इंटरैक्ट करना है. Tell me something. Melatonin, very good. Tell me something about melatonin.
We laugh a lot, so we produce melatonin, but we cannot sleep. So
You are unable to sleep. For sleep also there are different kind of hormones. Melatonin is also called, but there is a set of hormones. So in human body, more than 50 hormones are there, right? So we do not know. One hormone is upscale, another is, so there is a balance of those kind of hormones. So who knows? You are not sleeping well because of certain other reason, not because of melatonin only. So we don't know. So this is a very complex network is going on inside the plant system and inside your body also. Nobody knows. Or you all have very beautiful what? I do not have. लेकिन मेरे को ऊपर वाले से कोई गरज नहीं, है ना? My wife and my daughter says I look handsome and smarter in this look than the hairs.
Thank you. So इसी स्लाइड पे खत्म कर देंगे. No issues, because जो मेरे को बताना था वो मैं बता चुका हूँ, है ना? So the concept I wanted to tell you, all the other, because for science, if you have to prove certain kind of hypothesis, you have to provide evidences for any kind of science, right? So in my case, there are a lot of evidences. मैं कुछ दिखाऊंगा, कुछ genes upregulate हो रही हैं, कुछ downregulate हो रही हैं. We have identified certain new genes which is associated with these two kind of phenomena. So वो सब दिखा सकते हैं. So those, because I know UG, PG and doctorate students are sitting here, right? So all of you are not very well versed with those kind of evidences. But those who are very well versed and who wanted to continue the things, my phone number and my other details are there, and my email ID is also there. So if you want to connect with me, I'm really try to help you, and one way or other way, if you want to continue here, because you all are very good budding scientists, I know that. So I'll stop it here, but I would like that you should share or you should interact some of your thoughts or some of your queries. If you have any kind of no question is bad in original sense. So whatever the bad question you have, even if you want to ask my name again, please ask. But interact, right? So thank you very much. Thank you. So I would like to interact, like she told me about, we discussed about melatonin. So right now, if you have any kind of thing, anything related to my topic or other topic, we can talk about. Just 2-3 minutes, because my other friend is waiting. He is also one of the, he is not also, I am not at all. He is one of the best orators. I know that. I will introduce him. But first of all, tell me if you have any queries or questions.
Sir, did you find any plant which show both?
That's what I told you. We have identified certain germplasm also, certain genotype also, as well as certain real population also. So we deposit it into the MBPGR germplasm conservation system also. So we identify, because I told you there is a recombination during the evolutionary phase, right? And so some of the genes or some of the chromosome interact with each other, and they provided the better part of this SRM, stem reserve mobilization, and better part of stay green traits. Both combined together, and they were providing the best genotype which we were interested in, right? So we got.
Is there any estimation of how long the strain?
We did that particular part also. We did that. Very good question. Basically, I'll say like that. So from we were having the life cycle of wheat plant up to 110 to 120 days, but we got in those 135 to 140 days, up to 135 to 140. So 10 to 15 days delay in the process of senescence, we got those kind of germplasm. And that did a marvelous thing for the plant system, because we want to have the maximum benefit from the plant for our benefit. We don't care what is going on, but we are compromising on the basis of quality. So quantity we are getting for the shorter duration variety, but the quality we are compromising on those. But these kind of germplasm or real population we got, we never compromised on the basis of quality. We did those kind of experimentation also, where the quality was also up to the mark, as well as the quantity was high.
Thank you, sir, for the marvelous.
Thank you, boss. Thank you. Okay, no question? Okay, then I'll, if you permit, yeah, please. So I will request my very good colleague,
Dr. Naved Sadiq. So he is very good friend of mine. You know how we were friend when we were in IRI. Both are in IRI. We are both very good cricketer. And still I am. He is not. I am. No, he is not because he is not playing. I used to play every Sunday. Every Sunday I used to play till date. Till date. Because I am lucky enough to got these kind of work. And he is very good orator and very good poet also. Very good poet also. And he is class apart, excellent teacher. So I don't want to waste time. So Dr. Naved, now floor is yours. You have 20 minutes with you, and stick with 20 minutes only. Another 20 minutes we will take. Right? So I think you will enjoy this recipe also. Okay.
Good afternoon, Dr. Ajay, and thank you, the organizers, for giving me this opportunity to speak. And we had a lot of good science, more and more into less and less kind of microcosmic and microbiology and biotechnology and genes, and wonderful lectures from Chakrabarti sir and Prashant Mishra sir. They were really a treat to hear. Now, from this microcosmic world into a macrocosmic realities of sustainability. Everybody these days talks of the sustainability, more so when the climate change, global warming, and things like global dimming are being talked about. And we are on the crossroads, on the dangerous crossroads, where we see a lot of dangerous things upcoming. A lot of wonderful discoveries are also coming. Scientists telling that there is nothing like time, and there is something like negative time. There is that the core of the earth is slowing down. Some say it has already stopped. With the climate at risk, but the technologies at the helm of the things, there is where the efforts for sustainability have to be looked into in multi-directional, if not just bidirectional ways. Sustainability, if you go to the definition of agriculture, ecology by principles of ecology by Townsend, it is just repeatability of the action. To put simpler terms, if you drink most pure poison, you might die. I say, I must die. You're all young ones. Example was wrong. So I'll die, I will waste. And if you reduce the poison, so that exactly is the definition, simplified. So any action that can be repeated maximum number of times ends up being the most sustainable. The definition of sustainability is that sustainability is a choice between self-interest and the systems. If you choose system over self-interest, that as they say, that the charity begins at home. There the sustainability begins. Now, in the area of agriculture, since the technologies are increasing, we have reached a stage where we can grow crops worth 100 acres from just 1 acre. Now, this is the time actually to relook our options and opportunities towards the sustainability. Life is a game of choices. So if you choose to the run-of-the-mill technological intensity of producing 100 acres worth of crop just from 1 acre, or put rest of the 99 acres to better use, or in fact no use, for the nature to let us heal.
So we are at such kind of crossroads. We have seen divergent spectra of agri-tech adoption in the country, from low productivity due to small holdings, lack of education, irrigation, mechanization, climate dependence, institutional credit deficiency, unprofessional approaches, unorganized markets, lower risk-taking abilities among the farmers, and last but not least, the least export linkages available to most of the farmers. With the burgeoning population, the land holdings in the country are so low that we are virtually forced to intensive cultivation. Lower the size of the land holding, more intense are the wastages and intensities of compulsions of profit earning for realizing the marketable yields for sustaining the families. So to take advantage of these, exact reverse is the higher end of the technology, where I am working in the Center for Protected Cultivation Technology, that itself is one of the upper ends of the technological advances in agriculture, where you see greenhouse agriculture. That is protected cultivation. You all know fallouts of the, I will not go into detail, these are most of the general things, just wanted to highlight that fallouts of intensive cultivation include the soil degradation, soil erosion, water depletion, loss of biodiversity, and you know we are second most biodiverse nation. So if anybody is going bankrupt in biodiversity, we should not bother much, maybe. But India, ये मुशायरा तो कल है, लेकिन मैं ऐसे ही बता देता हूँ कि वो जो उसको गंवा बैठा है, मैं भला कैसे गंवा दूं उसको. So anybody can afford to lose biodiversity, but not a country like India. We are a big repository of biodiversity in the world, and depletion to that is a huge loss to the whole world rather than us. Then things like greenhouse gas emissions, increased pest resistances, leading to higher and more intensive use of the pesticides. Fortunately, the government is putting a lot of effort on organic cultivation. But now we are at again the crossroads where if we do less pollution, the global dimming takes over. People say that our global warming has been around 1 degree centigrade in 100 years, but in actuality it is much more. And the countries who have resorted to very intensive environmental cleaning have suffered heat strokes worse than the ones who have not done. It means we are at a choice of devil in the ditch. Either you face pollution and things like suspended particulate matter, or you face more global and actually warm things. It's going in reverse. It's going in reverse direction. Okay. No.
It is going.
Yes. So our country, we are having different kinds of greenhouse structures for taking advantage of biotic and abiotic stresses, controlling them, increasing the productivity 5 to 10 times, and by virtue of verticality, we can increase the growth again 10 times, so you can go 100 times from one point. Then better quality, off-season production, minimum use of pesticides, amenable to organic and safe cultivation, and suitable for peri-animal farming and better agri-entrepreneurship and economic propitiation. Different kinds of greenhouse structures are used. If you want to go into detail, you will have to read, but I'll just introduce, depending upon the region, the climatic factors, the abiotic factors, biotic factors, the crops, the seasons, the regions, you select different kinds of structures for different choices. So here, whole basket of structures. I showed my email if anybody is interested in going into those kind of details. It's most applied aspect of agriculture and very, very relevant these days, at least in the wake of the climate change. We, through the protected cultivation, can increase, as I say, 5 to 10 times more yield. If you want to go into detail, since most of you are not from agriculture background, you're mostly from botany kind of things, so not largely into applied things, but you can take the cues that we can take cost-benefit ratios much higher than the open field cultivation. And we can increase not only the quantity, but also the quality of the produce and getting the better prices. But there are several issues. There are several issues in context with the protected cultivation. There's a design of the technology. The companies have to provide the healthy planting materials, soil management, better protection practices, and better engagements like food safety management systems, and particularly in the post-COVID era. Now, most of the people are seeing the YouTube and all, and most of the people are interested in hydroponics and all. We get 5-6 queries every day in our office to increase hydroponics and things like that. But there are a lot of challenges. I'll highlight some of them. The key challenges are that the selection of the type of the structure, because it involves a lot of investment, although government is giving subsidy. Training and technical competence of the workforce who needs to be deployed there, the testing facilities, quality inputs, the market sensitivities and environmental hazards that you have to take resort to. If you are using a lot of plastics, then you have to be responsible. So in the wake of this intensive use, we are supposed to resort to good agriculture practices. Now, I'll tell you a small couplet like thing. कहते हैं खुदा जब हुस्न देता है नजाकत आ ही जाती है. So good agriculture practices are like that only. If you have good affordability, good accreditability, good availability, then you choose. If you are hungry, then you don't choose. If you are rich and you can afford, then you buy quality, buy variety, buy imported, things like that. So in Europe, when people have a lot of good affordability, and when the global trade opened somewhere in 1992, they started getting agricultural produce from all over the world, and they were really worried as to what they were eating. So when they started worrying about, so they set up a commission wherein all the stakeholders in agriculture and science and commerce, they got into play and they developed things like, so I'll not go into detail good agriculture practices, but it has 4 pillars, that is economic viability, environmental sustainability, social acceptability, food quality and safety. Instead of remembering this big definition on good agriculture practices, I have my own simple definition, that it is responsible and responsive production system. Responsible because you are responsible for the farmer who is producing for its profitability, responsible towards the society at large for the food safety, and responsive to the government compliances, the social compliances, the export compliances, market compliances, and all that. So now, if you go to the market and pick up suppose a cabbage, and can you know which chemical was sprayed, where it was produced, how it was done? So to ensure that, good agriculture practices resort to 14 major principles. The first major principle is traceability. You can put my name and gap, you will get a bulletin, you can read it plan. Don't need to worry about learning these things. It's already on the internet, my bulletin, you can read it. Traceability is that you can ensure if there is an arrangement between the producer and the seller that you will produce the crop as per such and such guidelines. You can use this pesticide, you cannot use this pesticide. If you want to use this pesticide, you have to use minimum this number of days before the harvest so that the harmful chemicals do not reach the table of the person eating. Yes, I'm finishing.
2-4 मिनट की बेईमानी करूंगा क्योंकि आपने बैटिंग ऑर्डर बदला है. You used to bet lower than me. Because he was a good hitter of sixes, so he come down. Anyway, the other principles like record keeping, varieties. So you record everything, where it was produced, who produced it, what chemical was used in that place, which worker was there, and whether worker safety was not there or not there, what was the microbial health standards and all that. All that has to be recorded under the production system. And that is how you can ensure a healthy, safe food at the profitable and keeping the environment sustainable as well as the economy profitable and the producer happy and the eater happy. So that is how, and responsive means you have to address to the compliances, to the complaint forms. So now climate smart agriculture, because of the stresses due to climate change and all, and increased technology, now climate smart agriculture using different kinds of sensors for electrical conduct, for example, water, electrical conductivity, the pH, the nutrients, all are measured and given in a controlled temperature, controlled humidity, controlled the detection of the inputs. These methodologies are like coming up day in, day out, and we have all kinds of systems like hydroponics, aeroponics, geoponics, aquaponics, many terms. Aquaponics, where you use fish excreta to be used as input into the. So at our center these photographs are from our center only, Center for Protected Cultivation Technology. Anybody who is interested to visit, particularly the students, they can come and see these facilities. We are getting at least 1 lakh visitors at our center every year. These are the aeroponic, and this is geoponic tomato on the left side, where solid soilless medium is being used with controlled irrigation fertilization phase. So climate smart agriculture is basically controlled environment with precision farming, biodata, data-driven decision-making systems, including big data analytics, automation of machinery, including robots, drones, micro drones, sensor technologies, and the Internet of Things and artificial intelligence, and sustainable agriculture practices, including good agriculture practices and market opportunities linked with these systems. So sustainable farming, actually the United Nations have given 17 sustainable developmental goals for the whole world to follow, where not only the prosperity, profitability, sustainability of the system, life on land, life on water, the economics, the gender issues, all have to be adhered to. It's like virtually utopian system into good of everything. Although it might not be possible, but yet we have to progress and proceed towards that scientifically and in a very sustained and methodical manner. The things like this organic farming, agroforestry enrichment, biodiversity enrichment, crop rotations, polycultures, regenerative agriculture, integrated pest management, hydroponics and vertical farming and precision agriculture have to be used in tune with overall ecology and economy as per the region and the systems to stay sustained. So that is my last slide, sir. So if we are doing climate smart agriculture, I did already say one thing, that if we are technologically equipped to produce crops worth 100 acres from just 1 acre, but we have to make systems energy efficient, cost effective, and greenhouse systems which are high intense cultivation with high investment. So we have to make farmers sustain. Now around 70% farmers are actually failing in the greenhouse technology, not because the technology is bad, but because they cannot sustain the intensive technology, so they have to be support system. So traceabilities, promotion of the sensor-based IoT and AI brain-based systems to streamline systems, minimize the losses, maximize the profits, and adhere with this. Then the sustainable agriculture systems also there is time, opportune time, and right time to actually
go in for compensatory mechanisms for the industry. Suppose there is a pesticide industry, and if there is a mechanism just like carbon credits, that if they have to produce one chemical, then they have to produce, say, two green chemicals, two green molecules, so that over a period of time, the harmful pesticides, or which are, if not immediately, after a few years, they may be replaced, and reciprocal compensation will be the one method include, and besides biological control mechanisms and vigil on quality and compliances of these things. So, and at the end of the day, nothing succeeds like success. The farmers, most of them, they do not do it for the social cause, they do it for sustainability and for their own families. So ultimately, the market mechanisms, I want to sensitize this is a time of big data, and this is a time of cyber fraud also. So at the end of the things, I often say to the policymakers that if all the data of the farmers, if it comes on one desk, then they are really vulnerable. That data has to be really, really very safe. Otherwise, some big player will play with the prices of the commodity which is available in one region and not available in the one region. So they could play with the distribution mechanisms. It is all really fraught with risks and dangers. So the technology, like always, just like artificial intelligence is used these days, you are seeing today itself, I got a call on my phone from wife calling. My wife visiting here. No. It's not a normal call. It's not a WhatsApp call.
Lucky boss.
But why?
You are lucky.
So these artificial intelligence and things like cyber fraud, they have to be seen with big data analytics in a very, very sensitive and very, very balanced and cautious manner. So caution is the key for today. This is the rose from my garden. This is quite sustainable. Thank you very much. It was just I wanted to like interact with the students after a long time, and time is less. Otherwise, we could have talked a lot. Dr. Ajay Arora, he virtually sacrificed his speech for mine. Thank you very much. But I must appreciate the way he interacted. The science, the real scientist is the one who simplifies the science rather than complicates it and confuses it, and at particularly this budding age. So thank you very much, and thank you all organizers. And if there are any questions, I'll be more than happy if I could answer.
Thank you, Dr. Nave, for a wonderful exposition of climate smart agriculture. I think this is the key which is going on in agriculture. So you take his email ID or the phone number from the organizers. So whenever you want to come to IRI for seeing this CPCT facility, he will show you each and every one there. It is difficult to come to that particular place because a lot of visitors are coming, but for new people, he is always open. So most welcome to IRI.
Thank you, Dr. Zainul Abidin in particular. Yeah. We are mates since 15 years of age.
Yeah.
When I was 15, from 11th class to date.
So, okay, thank you. Thank you, Dr. Nave, because we are running short of time this morning. So now I will introduce Dr. Iqbal Khan. So in-house person, our own person. So the budding assistant professor, but going to be soon associate professor, and one of the best biotechnologies I have seen since last 5-7 years. So I would request Dr. Iqbal to speak on rare earth element and plant hormones. Again, the plant hormonal interaction and how the plants are responding to those kind of effects. So he will speak on. So stick to your time, around 20 minutes.
Yes, sir. You can use that one.
Yeah. Pointer.
इससे ये पॉइंटर है, इससे स्लाइड्स अप एंड डाउन होंगी. Yes. So, PowerPoint found problem with the content. Okay. So can you use your pen drive? Actually कुछ corrupt हो गया छुआ है. So yes. So I'll play this from here. Huh? So ये add खत्म हो जाएगा. This file is not open.
मैं तो लाया था, काम तो कर रहा था. मैं तो लाया था, काम कर रहा था. लेकिन नहीं आया, मैं लैपटॉप में भी जोड़ा है.
कोई नहीं, ट्राई करते हैं.
रिपेयर करना. अब ऐसे करता हूँ, मैं भी ऐसे बात करूँगा.
ठीक है, जस्ट जो सर का वहाँ देखो, दूसरे लैपटॉप पे देखो. ट्राई कीजिएगा.
काम नहीं कर रहा, लैपटॉप खुल नहीं रहा पीपीटी.
ये डुप्लीकेट कर दीजिए इसका. फिटिंग क्या है?
मैं लास्ट मोमेंट पीपीटी फाइनल करके लाया, यहाँ चेक नहीं किया.
Not working.
पीपीटी इज नॉट.
जब तक ये सेट कर रहे हैं, कोई जोक वगैरह सुना दो यार.
सेम.
So something wrong with the PPT. So leave it speak like me for 5 to 7 minutes.
Okay, I can speak.
Yeah, I know.
Okay. Please, उसमें रख दे भाई. Okay, basically all our students from BSc and MSc and PhD, I think, right? So all are from the biotechnology department. Mostly are from the biotechnology department, and some are other departments also. So sorry for that inconvenience. I prepared very good slides for you people. I was knowing that most of the audience are going to be a student, the initial students for the research. So the PPT was according to you, but unfortunately it's not working here. And for the first time, I'm not able to make a backup, and it failed. So lesson number 1, always you should have backup. Don't forget, right? So my topic was turning plant responses with rare earth elements and plant hormone. If you are going through that, if you can listen me, so you will get some lead in the research also, right? So if you want to do good research, first thing you should have some patience. Otherwise, you are not going to be do good research. Note down, please. So 3 keywords in my title you can see. First is plant response, second rare earth element, and third is plant hormone. So everything we are doing here and we have some response, either positive or negative. So in the same thing, in the human body, we have the positive and negative responses to anybody, right? Anything, antigen, antibody, and whatever you are taking the meals and all have the some response. In the same way, plants has its own responsive system for everything, either for the climate, either for the mineral nutrition, either for the soil types, either for any conditions which are you going to give. So my crop is basically wheat and rice. These all are the staple crop and very economic, and everybody is eating in the world. And we are facing a very drastic changes in climate. All we are knowing about the climate change, and all we know how the environmental degradation we have done throughout the world, and how these are going to be impact on our food system, because we are urbanizing day by day and shrinking the agricultural land day by day. For example, now we have Greater Noida. Greater Noida is all where one time it was all agricultural land, where the crops were growing, mustard, wheat, rice, these all crops were growing. And now we have lots of societies. So we are increasing in population, shrinking the land, the agricultural land, and we need more foods because we are increasing in population. We need food under that climate change condition. So in that response, the plant scientists are trying to tackle up these harsh conditions for the agriculture production. The all cumulative impacts, all cumulative resources we are using to protect our crops. Rare earth element I choose just before 3 years when I was reading an article on green revolution and its impact. It was recently published in 2021, and it was mentioning that the green revolution was tremendous in India, and it has a lot of impacted on the farmers, on uh, people to get the food at the cost of chemical fertilizers used. So chemical fertilizer was used extensively without calculating the risk factor, the suspend factors of the soils types and its fertility. And now we are facing a very crucial and key problem in the agricultural land, that we have maximized the ammonium accumulation, the end source. And due to that, the deterioration in the soil is going on, and it is going to reduce our crop production. So we need some type of other fertilizer which can help in growing the plants, and the yields should be sustained for the whole population. In that way, I was again reading some further literature, and I found a term, rare earth elements. I hope you are knowing that we have F block in the periodic table, and all F block elements are the rare earth element itself, saying that they are very little in percentage, and they are also a core component of earth crust, right? So many countries are mining coals and all, and they are extracting the rare earth element, but it's all are going to be waste. But in China, China is a very beautiful country in any aspect of the research, in any aspect of the lifestyle. They are using that waste material as a fertilizer, the very natural fertilizer which have no any consequences till they are using since last 40 years, and they are producing the crop production. So that clicked me, why we are not going to test here at Jamia Hamdard, right? So I focused on the lanthanum, one of the rare earth element, and plant hormone research is my specialization, so I put it in the hormone also. So that lanthanum is very fantastic in response. It is two-edged sword. You know, two-edged sword, do tarfa talwar, dono taraf se kaate, right? It means that it has some positive and negative effect. Everything in the universe has its own two effect, positive and negative. If you are going to feed any of the friend, suppose biryani, more than he or she wants, then there will be some stomach pain or anything else can be happen, right? So everything is needing at the optimum level. So it has the toxicity effects also on the plants. We tested that from the gradient. We just published in the Physiology of Plant Journal, wonderful journal. You can go through it. So that aerial earth element lanthanum responses in two ways, the toxic and beneficial element. And again, we have the plant hormone that is known as abscisic acid, ABA. This is commonly known as the stress hormone of the plants. When plants face some stress, the concentration, the production of the ABA shoot up. It means that they are responding with the stress condition. But we are not going through the stress, but the optimization of that ABA level will be beneficial for the plant growth and development overall. The whole metabolite profile was tremendously changed due to the rare earth element response, and that was accumulated towards the grain yield. Grain yield. Every farmers need more grain yield. That's why we are feeding so many kinds of the foods. We are sustaining that grain yield. So rare earth elements sustain the grain yield, and in the response of ABA, it was found that whatever the toxic impact of that rare earth element, the lanthanum, was minimized. Minimized means that the tremendous effect, the worst effect was minimized, the lesser. And it was interacting between the calcium, because lanthanum is also known as the calcium, super calcium. It replaces the calcium. If calcium's deficiency is going through the plants or in the soil, the lanthanum can supplement it. And calcium is one of the essential mineral nutrient. So this is three triangle story, which I was going to tell. Okay, so third point is that whenever you are facing some challenges, you should make some plan how to cope up. If you have a very tough question paper, then you cannot leave the examination hall, right? Otherwise, you will be failed. So complications in the agricultural production is very tough, and this is opening a very good door to us also. How? You can solve some problem, you can get a very good lab, you can get a very good scholarship, we can get a very good papers, patents, and that will help you in building a career, right? So agriculture biotechnology is a field where lots of money is coming from the various agencies, lots of career opportunity we have, and here at the biotechnology department, sir is doing tremendous work. किसी को आइडिया है किसी को आइडिया है अब्दुल सर और he is the one of the pioneer plant biotechnologist in our India who worked on Artemisia. So we have a lot of opportunity in that area. I request all of you students, think seriously for your career. If you are thinking that 1 or 2 years is okay, I will go and roam and round and then MSc is completed. Now what? You will be roaming around the PhD 4 or 5 years, then what now? So lots of opportunity in agricultural biotechnology. You can talk with me and sir. I can suggest. So this is the only I want to say. Thank you so much.
So at last, we should thank the organizer, Dr. Hamza Abdi, Dr. Monica, Dr. Javed, all the group, all the organizing committee, which they successfully completed the day one of this particular conference. I suppose another 2 days are there, sir? Another 2 days are there, sir. Hopefully the team will surpass that particular level which they made last year. I think successfully. So thank you very much, sir, for inviting all of us here. And thank you, Dr. Javed, for helping us in organizing this particular.
Okay, thank you. Thank you so much for nice lectures. Now this is the time of facilitation. Now may I request to our chairperson to facilitate our one of speaker, Dr. Naresh Sabi sir. Please, sir.
सर ये बहुत अच्छे बनवाए हैं आपने, बहुत अच्छे बनवाए हैं, रियली
Now may I request Professor Azad sir to facilitate our speaker, Dr. Arun sir. Now may I again request to facilitate Dr. Azad Khan. Now may I request Coordinating Secretary Professor Azad sir to facilitate our chairperson of this session.
हो गया बॉस, दो ले जाऊंगा।
Yes, definitely. Yes, yes.
I would now request chairpersons to felicitate Dr. Javed Ahmed with the moment.
आपको तो मिलना चाहिए।
Sir, now I request all of you and Nagit sir for a group lecture. Yes.
अरे यार ये बेचारे ये भी तो आ रहे थे, आ जाइए आप भी आ जाइए, आप भी आइए, सब लोग आ जाओ, आइए आइए आप भी।
Monica ma'am, you are invited on the stage. If you are here.
Thank you.
So that brings us to the end of day one of the conference. Thank you for your participation. We look forward welcoming you tomorrow as well in archives, that is Hakeem Abdul Hamid Auditorium at 9:30 a.m. sharp. Thank you all again.
So tomorrow we will meet you again at archive point.
ये रिकॉर्ड हो गया।
देखो जी, यहाँ से हमने स्टॉप किया, रिकॉर्डिंग हो गई स्टॉप। अभी रिकॉर्डिंग हमें मिल जाएगी जब हम चाहेंगे तब।